Meera.
சொல்லுங்க பிரியா.
Ready ma? Vinayaga sir. Vinayaga sir.
Yeah, ready sir.
Yeah, okay. Meera, you can start.
Yes sir. Assalamu alaikum and very good morning to one and all present here. Am I audible, sir?
Yeah, I'm getting you. You're clear.
Yeah, this is S. Rasul Mira, faculty of AIB and MC of this program. A very warm welcome to national level webinar FDP on vaccines and cryptogamic biodiversity. Now, I would like to call our association president, Dr. I. Niaz Ahmed, sir, to give welcome address and introduction of the chief guest.
Good morning to one and all present here. It gives me immense pleasure to be associated with you in another webinar. Wholeheartedly, it gives me a pleasure to thank and welcome the various stakeholders of this good cause. In absentia, I would like to thank Dr. Madhusudanan, our association secretary, Dr. Rao. In absentia, from the inception, my PhD gate, Dr. T.P. Shastri sir was associated with us. Now, I would like to welcome the chief guest of the session 1, Dr. Vinayaga K.S., Assistant Professor in Department of Botany, Sri Venkateswaramana Swami College, Vidyagiri, Banfal, Karnataka. And I would like to welcome the association member, a prominent member of our association and the organizing secretary of this webinar. I would call her as Dr. Rasul Meera because she is going to defend her thesis due course of months. And I would like to welcome various stakeholders as association members and other fellow participants of today's webinar. Yeah, welcome you to all. It's my duty to introduce today's chief guest, Dr. Vinayaga K.S. Presently, Sir is working as assistant professor and head the Department of Botany, Sri Venkateswaramana Swami College, Banfal, Karnataka. Sir has wide range of educational experience. If you talk about the educational qualification, Sir has qualified PhD and MS in Colombo University in the domain of applied botany. In addition, Sir has MA Sociology. If you talk about the additional qualification, he has various diploma holder, PG diploma in medicinal aromatic plants, PG diploma in genetics, PG diploma in human resource management, PG diploma in Kannada journalism, etc. Sir his various portfolio, he worked as employment history. He was a JRF, Junior Research Fellow. He was a lecturer and guest lecturer and assistant professor, and at presently he was a head of the department. Sir has also chaired as principal position in Coimbatore First Grade College, Shikaripura. His honors and awards, Sir has get various awards from 2008 till 2023. Some of the awards include DST Young Scientist Fast Track Project. I feel happy myself. I also awarded by Government of India with the DST Science and Engineering Research Board Young Scientist Project. Sir has got 2020. He was awarded with Young Scientist Award by Nature Science Foundation, Coimbatore. He also awarded as Young Achiever Award by Institute of Scholars, Bangalore. Sir's projects include various funded and non-funded project he was completed. While I'm reading his profile, I feel happy and point out to you, he was prepare a checklist of Karnataka legends and add the data to World Legends checklist database. It's give you an example of his social relevant project. He has completed 18 number of projects. He was membership in various association. We are feel happy and proud. He also associated with us in our association. He was a member of 20 different NGO. As a resource person, he was chaired various places, Shimoga, Udupi, and various conferences and seminars. SAR has organized various workshops, seminar, and DST inspired programs. He was organized as a camp officer supported by DST in the year of 2016. His research guidance include 85 BSc projects and 15 MSc projects and 1 PhD project. PhD scholar is working under him. Review committee member. He was a review committee member in various portfolio and various journals. The list goes long. His publications are various national and international journal with good impact factor. The number of publication is 111. His popular articles include, which was published in various good impact journal. His book chapter and books published, the number goes around 5. SAR abstract present and published in various conferences. The list goes and the number is 47. The conference, symposium, workshop attended in various places. The number is 45. With this short bio, I would like to welcome Dr. Vinayaga sir with us. Sir is going to enlighten us with his presentation. Before, we have 7 different presenters. They are going to present their research views related to plant biology. Sir is going to act as a jury for this session. With this introduction of chief guest and welcoming the gathering, now I wholeheartedly welcome Dr. Vinayaga sir with us. Welcome you, sir.
Yeah, thank you, sir. Thank you, Raj.
Now I call our organizing secretary, Dr. Rasul Mira, to take over the session for the presentation of the students. I request her kindly read the name of the participant. If they are not available, kindly give them a chance at the end. Let them to present their views. Yes, Mira.
Yes, sir. Yes, sir. Thank you, sir. Now I will start with instruction to the participants. Kindly listen. Only 5 minutes allowed to present your paper. Participants will be called according to slot numbers. After the presentation completed, the other participant can ask questions if you have doubts. The left out participants, you can do your presentation finally. When asking doubts, kindly ensure that the mic is unmuted condition. The final decision will be made by the judges. Now participant can do your presentation paper one by one. Number 1 is Sujita from Arcar College from Tamil Nadu. Sujita. Sujita, ma'am.
Sujita is here. Sujita is here. Here, she is in the webinar. Miss Sujita, okay. She is not getting us. Miss Sujita.
So shall we call next person?
Yes, yes, yes.
Yeah, T. Gayatri from Arcot C. Mahalakshmi Women's College. T. Gayatri.
Yes, ma'am. Yes, ma'am.
Are you ready to present, ma'am?
Yeah.
Yeah, you can share the slides now.
Good morning to everyone present here. We are from Arkat Sri Magalashmi Women's College, Villapakam, Department of Biotechnology. We did a team project. I am Gayatri, along with S. Indumadi and S. Sujita. Today I am going to present a project. The topic is screening of phytochemical and antibacterial activity of different fruits and vegetables. I am divided by my presentation into 5 parts. There is aim, objective, materials, and result. Now I am going to review the parts. Aim to determine the phenolic components present in different fruits and vegetables and evaluate the antibacterial activity of extract. Objective to prepare the solvent extract of fruits and vegetables and perform phytochemical analysis and determine the antibacterial activity of different extract of fruits and vegetables. Materials used for this project are grinders, 70% of ethanol, and Muller-Hinton agar medium, chemical.
Gayatri,
Yes, sir.
sorry for the interference. Kindly twist your mobile phone in the horizontal instead of vertical. We can able to see the slides.
Okay, sir.
And now it is good, ma. Please proceed.
Okay, sir. We start from first itself, sir.
Yeah. Proceed.
Good morning to everyone present here. We are from Arkat Sri Magalashmi Women's College, Villapakam, Department of Biotechnology. We did a team project. I am Gayatri, along with S. Indumadi and S. Sujita. Today I am going to present a project. The topic is screening of phytochemical and antibacterial activity of different fruits and vegetables. I am divided my presentation into 5 parts. There is aim, objective, material, method, results, etc. Now I am going to review the parts. Aim to determine the phenolic components present in different fruits and vegetables and evaluate the antibacterial activity of extract. Objective to prepare a solvent extract of fruits and vegetables and perform the phytochemical analysis and determine the antibacterial activity of different extract of fruits and vegetables. Materials used in project are grinders, 70% of ethanol, Muller-Hinton agar medium, chemicals, filter paper, etc. Method: the sample are lemon, banana, tomato, carrot, beetroot, and guava. The preparation of extract was: the sample was washed thoroughly under running water and cut into small pieces, dried in oven at 60 degrees Celsius overnight. Using grinder, the fruits and vegetables were homogenized into powder. 5 grams of dried sample powder was soaked with 50 ml of ethanol for 48 hours. The extract was filtered. Then we have to perform the phytochemical analysis. Phytochemical analysis is a bioactive component in fruits and vegetables. The materials were analyzed after the extraction with ethanol. The tests include alkaloids, flavonoids, phenol, tannins, terpenoids. There are more than 10 tests are in there. Then the application. Antibacterial activity is done by well diffusion method. The microbial inoculum over the entire agar surface, then hole with diameter of 5 is punched with sterile tip and desired organism is spread on surface of the Muller-Hinton agar medium with sterile cotton swab, and the volume is 25 to 100 ml of extract solution introduced into the well using the pipette. Then they place for incubator for 24 hours for 37 degrees Celsius. Then result. The result were observed. The phytochemical analysis is done in 6 samples. It gives a maximum positive result due to color changes. Fruits or vegetables contain the phytochemicals. We can consume the fruits and vegetables. It can able to stop and prevent from various diseases. Then the antibacterial activity is carried out agar well diffusion method using the E. coli and the Staphylococcus. Both organisms are commonly a disease-causing agent. The clear zone formation indicates a test against a test organism. It can able to finding the antibiotic will be most effective in treating your infection. That's all about our project. Thank you for giving this opportunity to present our project.
Now, yeah, it's very excellent presentation, ma.
Thank you, ma'am.
Yeah, now I call up Mr. V.R. Pradeep sir from Savita Dental College.
Good morning, ma'am.
Yeah, are you ready to present now?
Yes, ma'am.
Yeah, you can proceed it.
Ma'am, is the screen visible, ma'am?
Yeah, visible only.
Is my voice audible, ma'am? Sorry to
Yeah,
ask
yeah,
this.
everything okay, fine. You can
Yes, ma'am.
start.
Okay, good morning, one and all present here. And my research title is about role of antioxidant vitamins on the expression of HNF1 alpha protein. HNF1 stands for hepatocyte nuclear factor and sterol regulatory element binding protein 1C in PCB-induced experimental diabetic rats. Okay, antioxidant vitamins. What are these antioxidant vitamins? Generally, our body needs two basic vitamins, which is vitamin C and E. And these two vitamins are predominantly called as the antioxidant vitamins. And I have said you two proteins, the expression of two proteins. One protein is HNF1 alpha and the another one is SREBP1C protein. Okay, what is the role of these two proteins in the human body? This SREBP, that is sterol regulatory element binding protein's role, is to maintain all the carbohydrate metabolism, that is the glycolytic pathway, oxidative decarboxylation, substrate level phosphorylation, then citric acid cycle, it is otherwise called as TCA cycle. So all the carbohydrate metabolism is being maintained by this sterol regulatory element binding protein. Then HNF1 alpha, what's the role of this HNF1 alpha protein? This HNF1 alpha is predominantly involved in controlling the genetic expression of the neuroendocrine mechanism. Neuroendocrine mechanism are the hormones which are synthesized both by the anterior pituitary as well as the posterior pituitary hormones. And HNF1 alpha, this protein is predominantly involved in the control of the neuroendocrine mechanism of the pancreas. Pancreas secretes two, and it is both endocrine and an exocrine hormone. Exocrine gland, it is a mixed gland, and pancreas secretes two important hormones, which is insulin and glucagon. And these two hormones are essential for the maintenance of proper blood glucose level. And these two hormones will have a differential level. I mean, they will deviate from their normal levels in case of diabetic patients. Okay, now my research is about the use of antioxidant vitamins. So we are going to synthesize a oral supplement which is going to consist of antioxidant vitamin C and E, and we are going to test it on the diabetic rats. Okay, whether rats will be naturally diabetic? No, we are going to induce the diabetes in those rats. And the rats selected for my research is Westar albino rats. You may ask me a question, why did you choose Westar albino rats for your study? The main reason behind choosing this albino rats is such that albino rats have a chromosomal number of 42 and their major histocompatibility complex, that is MHC, is more or less similar to that of the major histocompatibility complex of the humans. And you can ask me a question, you have done a research on the rats, whether have you got the permission from the committees and all. Actually, our college have a unit called as Biomedical Research Unit, Lab and Animal Center, where we have these kind of Westar albino rats and all, and it is being approved by the approval committee of the India also. And after that, so now I have to say that these rats are being with diabetes. How can we induce diabetes in those rats? As my title suggests, PCB-induced experimental diabetics rats. PCB refers to polychlorinated biphenyls. PCB is a toxic compound which can cause certain kind of harmful reactions in our neuroendocrine mechanisms. In the same way, we have a PCB compound called as arachlor-1254, which is also called as A1254. This arachlor-1254 can induce diabetes in those rats, and the same arachlor-1254 can cause diabetes even in humans. It is said to be a pollutant. It is a very strong pollutant, and it can cause severe neuroendocrine problems. Okay, now we have to test the oral supplementation, which consists of antioxidant vitamin C and E. We have to test it on the rats. Okay, in order to test it on the rats, these rats have to be with diabetes, and we are going to induce the diabetes with the help of the compound which is arachlor-1254, which is a PCB compound. Now we have taken 18 rats in total, 18 Western albino rats, which weighs about 150 to 180 grams. And these Western albino rats are divided into 3 groups: Group 1, Group 2, Group 3. Group 1 consists of the control group of rats. Generally, especially in vivo research, whenever we are going to describe about a particular drug supplementation which we are synthesizing, we will compare it with the control group. The control group can be a human if it is a human-based study. It can be a rat if it is a rat-based study, or else it can be anything. This control group of population won't get, this control group of population will be a stable group. That is, they have a normal regulation, they have a normal bodily mechanism, they don't have any deviation. That is called as a control group of rats. So group 1 is control group of rats, and it consists of 6 rats. And the group 2 also consists of 6 rats. And this group 2 rats are injected with Arachlor-1254 at the dosage of 2 mg per kilogram for 30 days. So I am coming to tell that these rats were kept for 30 days in a constant period of 12 hours daylight and 12 hours night light. So the first group of rat is a control group. So the control group of rat is stable. They are not diabetic. The second group of rat are getting induced with the diabetes with the help of the Arachlor-1254. So group 2 rats is going to be a diabetic rats. And group 3 rats are going to be a diabetic rats with the induction or the injection of Arachlor-1254. Along with the diabetes, we are also giving our supplementation which we are going to synthesize or which we are going to suggest. That is the vitamin E and C oral supplementation. That is the antioxidant vitamin supplementation, which is also given along with that the compound that is Arachlor-1254, which is inducing diabetes. So we have 3 group of rats. First is control. Second one is a diabetic group of rats. And third one also consists of 6 rats. And the 6 rats are diabetic. And as well as they are ingested along with the antioxidant vitamins. That is the supplementation I am suggesting. Okay. I told about you 2 proteins, which is HNF1 alpha and SREBP1C. Actually, generally, whenever a human is getting affected with diabetes, there will be alteration in the levels of these 2 proteins, which is SREBP1C and HNF1 alpha. When one person is getting considered with diabetes, or else he is getting diagnosed with diabetes, the SREBP level will increase. For example, in the graph I have attached, you can see that the PCB-induced group of rats. The PCB-induced group of rats is mentioned in green color tabulation, and the green color is said to be higher in the PCB-induced diabetic rats. In the same way, for human also, for whoever getting affected with diabetes, they have an increased level of SREBP1C. And you can see that, as I said, the control group of rats are stable. The control group of rats are said to have a normal range of SREBP, which is 250 picogram per ml. And once the third group of rats is given in blue color, once after the injection with vitamin C and E, that is the supplementation I am suggesting, that is the antioxidant vitamins. On administration of antioxidant vitamins, the SREBP values again decreasing back to the normal level, which is similar to that of the control group. So now the SREBP is increased in diabetic rats, whereas on our administration of vitamin C and E, it is getting decreased and it is getting back to normal level. In the same way, I said you another one protein, which is HNF1 alpha, which is hepatocyte nuclear factor, and it is essential for the proper neuroendocrine mechanism of pancreas, as pancreas is involved in the secretion of insulin, glucagon, which are the vital hormones for the proper maintenance of carbohydrate metabolism. So if one person is getting affected with diabetes, his HNF1 alpha proteins level will decrease. That is, I have shown with green color. And once after the administration of my oral supplementation of antioxidant vitamins, they are getting again back to the normal level. The HNF1 alpha is again getting back to the normal level that I have represented in blue color. And the control group of rats are already said to be in a normal level. And on our administration of vitamin C and E, we are referring that it is getting back to the normal level on our administration. Then, okay, whenever if a patient is getting diagnosed with diabetes, we will check 2 important things. That is the fasting blood glucose level and serum insulin. There are many tests for nowadays to determine the blood glucose level. It may be glucose tolerance test, or else nowadays we have a AccuCheck monitor and all. But whenever we are going to a diagnostic lab, they will definitely check us fasting blood glucose level. The fasting blood glucose level of a normal person will be less than 100 mg per dl. And for a pre-diabetic patient, it will be between 100 to 125 mg per dl. And for a diabetic patient, it will be more than 125 mg per dl, or else more than 140 mg per dl. Okay, in the same way, the control group of rats is having a less than 100 mg per dl of blood glucose level. Whereas if they are getting induced with PCB, the violet color ones, are said to have an increased FBG, which is above 150 mg per dl, so that the rats are diabetic. The same strata is observed in humans also. And when they are getting administered with the anti-diabetic drug, that is the antioxidant vitamins drug, again the fasting blood glucose level is getting decreased back to normal, and they are again coming back to the similar levels of control. In the same trend, you can notice in serum insulin level also. When the diabetes is there, the serum insulin level is increased. When we are treating with our antioxidant vitamin drug, the serum insulin level is again getting decreased. So, and the predominant mechanism, I mean the materials and methods we took here is the HNF1 alpha protein and SREBP1C proteins are undergoing ELISA, enzyme-linked immunosorbent assay, which is an antigen-antibody interaction. You may ask me a question, why did you do ELISA for this test? As we need to interpret the results in the form of the graphs, and we have to check or we have to interpret whether the HNF1 alpha and SREBP1C protein levels in all group of rats, that is in normal group of rats, in diabetic rats and diabetic rats which are treated with our drug. So in order to check the levels of these two proteins in all three group of rats, we did ELISA. And you can ask me a question, okay, I've induced the rats with diabetic compound, which is Arachlor 1250. What happened to the rats after that? As these rats are induced with diabetes and these rats cannot be further used for any other purpose, these rats were sacrificed at the end of the research. So total 18 rats were taken, divided into 3 groups, which contains 6 each. Okay, and you may ask me a question, okay, you are telling HNF1 alpha and SREBP are studied for this research. From which tissue you took this? We took the liver tissue, that is the hepatocytes of these Western albino rats. And these hepatocytes of the Western albino rats are first treated with triazole reagent. Triazole reagent is the principal reagent for the cleavage of the cell wall or else the cell membrane. Once after cleaving it, we have to go to the nucleus, and chloroform is used to separate the DNA, RNA and the proteins. So chloroform will separate the 3 things. We need only the proteins, especially HNF1 alpha and SREBP1C. So these 2 proteins can be extracted with the help of isopropanol and NH4. Okay, coming on to the discussion and the conclusion part, you can ask me a question. Okay, you are telling that the antioxidant vitamins, that is vitamin C and D supplementation, can control diabetes and it can bring us to normal blood glucose level, and you are interpreted in the form of the graph. What is the novelty of this research when compared to already used anti-diabetic drugs? We already use many anti-diabetic drugs like metformins, bisulfonylureas, bisphosphonates and all. The difference between those anti-diabetic drugs and the anti-diabetic drug which we have synthesized, which is also called as the antioxidant vitamins, that is vitamin C and E, we have synthesized is that vitamin C and E can also give an effect of antioxidative effect to our body. That is, they will give us free of the reactive oxygen species. Other further reactions like dangerous reactions in our body won't take place, as these 2 vitamins have vitamin C and E have antioxidative property. Apart from antioxidative property, vitamin E also have anti-sterility property, and vitamin E is also good for the skin. And vitamin C is essential for collagen biosynthesis. The collagen is almost present in all the connective tissue. It may be a fibroblast or it may be an osteoblast. In whatever thing, we need collagen as the basic material. So the currently used anti-diabetic drugs just concentrate to bring the blood glucose level to normal, and they will increase the secretion of insulin. Whereas the drug which we have synthesized and which we have tested in the rats as an in vivo research is said to possess anti-diabetic property. Apart from them, we also have antioxidative, anti-sterility, and it also provides goodness to skin, and it also have the advantage of collagen biosynthesis. As collagen biosynthesis taking place, a person can quickly recover, and there will be a healing rate, which will be increased healing rate is seen in case of vitamin C and D supplementation is given. And cataract is the greatest problem in case of diabetes patients. Cataract, and as vitamin C is given in the form of oral supplementation in these people, there are certain chances to decrease the amount of cataract in certain patients. So when comparison to already used anti-diabetic drugs, our drug which we have synthesized has certain other parameters to be considered also. They are good antioxidants, they are good anti-sterility products, they can prevent from cataract, they can give a increased rate of healing process also. So the conclusion is such that for sure we can use this antioxidant vitamins as a supplementation in order to bring the blood glucose level to normal, and we can suggest this as a good alternative for the currently used anti-diabetic drugs, and we have done this research with all the permission. It is an in vivo research. Thank you for giving this golden opportunity to each and everyone present here. I'm so happy to present this paper, and I also got this paper published in under Scopus in this journal. It is got published in this month of January 2023 in a publishing journal called as HIV Nursing. Thank you so much, ma'am.
Yeah, such a wonderful presentation you have given.
Yes, ma'am.
Yeah, wonderful presentation, ma. Any other participants have doubt? Hello. Any participants have doubt? Yeah, I think no. Yeah, next person get ready. Miss
Thank you
Huria.
so much, ma'am.
Yeah.
Meera,
Ah,
Meera,
tell me, sir.
the candidate itself, you can ask a question, I will respond. Like that, he itself writes the question and answer. That's why there is no right.
Yeah. It is very clear.
Sir, I'm so happy to present this in front of you people, sir, and especially in front of the chief guest. You added many things about the chief guest. He really stood as an inspiration when you were saying about the chief guest, sir.
Thank you, thank you. Dr. Vinayaga, sir.
Yeah, thank you, sir. Thank you very much.
Okay. Sir, we have some other participant. After you can proceed with your talk. Meera?
Now I call upon, sir, shall I call the participants?
Yes, proceed.
Yeah, Miss Huria like Ahmad Khan from Savita Dental College. Miss Huria? Yeah,
Am I audible?
Yeah,
Yes,
yeah.
ma'am. Okay. Good morning, everyone. Is my presentation visible?
Yeah, visible, ma'am. You can continue.
So good morning to one and all. Today, I would like to present a paper on the topic, the evaluation and comparison of the anti-inflammatory potentials of 3 plant-derived compounds, which is lupiol, piperine, and beta-cytosterol. This is an original study. So I give you a brief introduction on our topic. So first, coming to the term anti-inflammatory in our topic. So basically, inflammation is good for our body. It's a defense mechanism and it is beneficial because it protects our body from infection and injury. But suppose we have excessive inflammation and it's not acute, it lasts for a long period of time, that can lead to collateral damage. Now, what kind of collateral damage is that? It can lead to arthritis, neurological disorders, and even diabetes and cancer. So to prevent this, we have anti-inflammation that is mediated by medicines like non-steroidal anti-inflammatory drugs and steroidal anti-inflammatory drugs. But the problem with both these classes of drugs is that non-steroidal anti-inflammatory drugs will cause side effects. Like we have allergies, impaired renal function, stroke, heart diseases, ulcers, arthritis, even cancer. And steroidal anti-inflammatory drugs, they can also cause swelling, allergies, heartburns, vomiting, nausea, and all that. So we are going for more natural, more safer and cheaper anti-inflammatory drugs. Now here in our study, inflammation is related to protein denaturation. As we all know, tissue injury during the life, it might be referred to the denaturation of the protein constituents of cells or of the intercellular substance. So that is why here our anti-inflammation, we are going to be looking at protein denaturation inhibition. So the protein we have taken in our study, that will be bovine serum albumin. So there is a need to find safer and cheaper anti-inflammatory drugs. That is why we have chosen 3 plant-derived compounds, so that they are found everywhere and they are definitely safer and cheaper. Now, I'll talk about the 3 plant-derived compounds we have chosen, that is lupiol, piperine, and beta-cytosterol. So lupiol, we have a few sources of lupiol here. So lupiol is generally found in strawberries, olives, mangoes, and then piperine is basically your pepper. So it's like black pepper, white pepper, long peppers. As we know, during COVID, we all had corn cushions, soups, everything having pepper in it because of this potential. And then beta-cytosterol is found in walnuts, flax seeds, all kinds of nuts, and avocados. So that is why we are looking at lupiol, piperine, and beta-cytosterol. Now, the phytochemical screening done for lupiol, piperine, and beta-cytosterol has shown that lupiol is a pentacyclic triterpenoid, piperine is an alkaloid, steroid, and beta-cytosterol is a pentacyclic triterpenoid of the phytosterols. So all these three compounds, they are known to possess anti-inflammatory properties. Now, the novelty of this particular study is that even previously lupiol, piperine, and beta-cytosterol's anti-inflammatory properties, they've been established. But this is the first study that has been done where you're going to compare and evaluate all these three compounds anti-inflammatory properties, and we're also comparing it with our control which we're using, and that's called aspirin. So now I'd like to tell you about the materials and methods of this study. So first we have taken the sources. We have taken mango, we have taken pepper, and we have also taken walnut. So these are our three sources of the three compounds. Then we have extracted them. We have used ethanolic extracts. We have used it for the protein denaturation inhibition, which is done with a bovine serum albumin. So that assay was done, and then statistical analysis was done using one-way ANOVA and Duncan multiple range tests. And then the absorbance was measured at 660 nanometer by UV visible spectrophotometer. And after this, the percentage inhibition of the protein denaturation was calculated in percentage. So that is calculated using this formula: absorbance of the control minus absorbance of the sample by absorbance of the control into 100. So this was done in, this experiment was carried out in triplets. And as you can see, the p-value was considered lesser than 0.05. Only then it was considered to be significant. Now coming to the results. We have three graphs here. As you can see, first I'll explain you the legends. The x-axis represents the concentration of the plant extract that increases from 100 to 500 micrograms per ml. Y-axis, you can see the percentage of the protein denaturation inhibition that ranges from 0 to 100. Now we have these bars, right? So the red bar in all of the graphs, the red bar will represent the percentage of the protein denaturation inhibition by our standard drug or our control, which is aspirin. Then the blue bar in the first graph, that is for beta-cytosterol's protein denaturation inhibition, and the green bars is for piperine and lupiol in the next two graphs. So that will give you the protein denaturation inhibition of the other two. So each bar, as you can see, it represents the mean with or without a standard deviation of 6 observations. Now, what is this A, B, C, D that's written there? So A is basically comparing the protein denaturation inhibition with 100 micrograms of the previous value. So we have A is compared with 100 micrograms, B is with 200 micrograms, C is with 300 micrograms, and D is with 400 micrograms. Significance was considered at p-value lesser than 0.05. Now the results have shown that the protein denaturation inhibition of lupiol, piperine, and beta-cytosterol, it occurs in a dose-dependent manner, as you can see. And the concentrations were ranging from 100 to 500 micrograms per ml, and the maximum inhibition was still considered by the standard. As you can see, with the standard still has 90% of the maximum protein denaturation inhibition. That was followed by beta-cytosterol, which had 87%, then piperine, which had 85%, and then lupiol, which was 75%. But all of this, you have to remember that all of this was done for plant-derived compounds. So this is a very good result that we have got. And we have also done one more thing. This is basically your half maximal inhibitory concentration. So this is IC50. It's basically the measure which indicates how much of one particular drug is required to inhibit in vitro a given biological process. Here our process is the protein denaturation. So how much of the drug was required to 50% inhibit the protein denaturation inhibition. So lesser the half maximal inhibitory concentration, greater is the percentage of the protein denaturation inhibition. As you can see, our standard aspirin that had the minimum IC50, which means it had the maximum protein denaturation inhibition and is hence the most anti-inflammatory. So that was followed by beta-cytosterol with a value of 210, piperine with a value of 240, and lupiol with 320, showing that beta-cytosterol, out of the natural derived compounds, beta-cytosterol had the maximum anti-inflammatory efficacy. Now coming to the conclusion, all of these compounds which we saw, they were plant derived. So because of that, they're easily accessible, they're safer, they're cheaper, because they wouldn't give you allergies, and they wouldn't lead to neurological disorders like how the non-steroidal anti-inflammatory drugs and the steroids could cause. that would happen in the chemical compounds. So now we have done the basic study where we have just, this was in vitro and this was just with the extract. So later, with further in vitro, in vivo and clinical studies, these plant-derived compounds could quench the thirst of the herbal products in the herbal market. So it's clear that all of the three natural compounds, they have the ability to inhibit the protein denaturation. But here, what have we seen? Beta-cytosterol had the maximum inhibitory effect. As you can see, that was 90% for aspirin and 87% for the protein denaturation inhibition of beta-cytosterol. So that was it was very close and it was very good for a plant-derived compound. So then it was followed by piperine and lupiol. So all these three drugs can be, they can be recommended for the treatment of inflammation with further studies. But first, since beta-cytosterol had the maximum efficacy, that could be prioritized because it had a better efficacy. Now I just tell you the limitations and future scope of this particular study. Limitations is that because they are natural products, we don't really know where lupiol, piperine and beta-cytosterol can always be found. So they may not be found everywhere, and during culture also they might get damaged because they are natural and they might be friable. Now coming to the future scope, all of them can be recommended for the treatment of all these anti-inflammatory conditions. We have arthritis and also for diabetes and its associated complications also. But beta-cytosterol can be prioritized first because it had better efficacy. So that can go for more in vitro, in vivo and clinical trials. Thank you.
Such a wonderful presentation, ma'am.
Thank you so much, ma'am.
Yeah, very informative too.
Thank you, ma'am.
The other participants, if you have any doubt, you can ask now. I think no doubts from anyone. Okay.
Yes.
Thank you, ma'am.
Thank you so much.
Yeah. Next, get ready, Mr. Gajarajan sir from New College. Mr. Gajarajan sir. Mr. Gajarajan sir here.
Mira, I don't think he is available. Okay, Mira. Let me call our resource person of the day, Dr. Vinayaga sir, to present his presentation to enlighten us. As we received a feedback from the fellow participant, this much of workflow spokesperson with us. Now we call Dr. Vinayaga sir. Vinayaga sir.
Yes, sir. Now I will share my PPT. Just a minute.
Sir, excuse me, sir.
Yeah, Sarvesh.
Yeah, I'm in Berlin, sir. That laptop.
Ma'am.
Yes. I'm available, sir.
Okay, you do one thing. You wait after SAR presentation, you can proceed.
Okay, sir.
Miss Inbawali, your slot is next part. Regarding, hold on. One instruction. We have another spokesperson, Dr. Jagannathan sir from Pastor Institute. He is going to join us around 11 o'clock.
Okay.
After our first resource person, Professor Vinayaga completed, after the entry of the second spokesperson, the vaccine related presentation, Ms. Subham, Ms. Inbawali, Aisha Shabin will be present. Kindly hold on.
Okay.
Be with us. Thank you.
Okay, now I start, sir.
Yes, sir. Please proceed.
Yeah. Good morning to all, respected dignitaries and participants from the various colleges. First of all, I'm very thankful to Nia Samar sir for giving me an opportunity to interact with all of you. So in this today's my presentation, just I will overview what is the importance of biodiversity in Indian context. Just first we look at this picture, so you can realize what exactly the biodiversity. So the biodiversity is one of the very important component of our environment. Just it includes all living organisms. That may be the small microbes or maybe the larger evolved animals or plants, and all these things have included in our biodiversity. Just look at this picture so you can recognize what exactly the biodiversity that include all the whatever the living organisms existed on our earth. So when we take the just general account of this biodiversity, so whatever the life forms are present on the earth, the plant especially, because I am today focusing on the plant groups, so the about 82% of the global mass is made out by the plant group, and among the plant group, the 86% of them are occur in the terrestrial environment, and only the 13% of occurs in the deep water and other means, and only 1% are occurs in the marine environment. When we take the global biodiversity, the current existing is 1.8 million, but the scientists have predicted that around 8.7 millions of species occurs on the earth. And when we take the distribution of these different group of the organisms on the earth, the mainly majority of the organisms occurs in the tropical region, because they are very diverse in the environment in the tropical region. So that's why the majority of the whatever the living organisms are present, especially plant group, are reported from the tropical regions of the world. When you take the just statistics, so just look at this pie chart. So majority of the total biodiversity is represented by animals. In that, the many of them are belong to the insect groups, because the insect is one of the larger group of the living organisms. They are largely distributed in the world. And when we take the plant group, the mainly the dicotyledon plants are largely distributed on the earth. And these are the discovery patterns of the different group of organisms on the earth. So that maybe the virus, bacteria, fungi, or etc. Because whatever the known species of virus, just only we are know only 1%. Still now we are discovering various new species of virus and bacteria nowadays, because of in the form of diseases, or when it upcoming the various new diseases that may lead to the discovery of the new bacteria and viruses. But whatever the plant species group, so they are largely distributed in a different part of the country. So based on that, they divide into a biodiversity hotspot regions. There in the world, there are about 36 biodiversity hotspots are there. When we take the India, there are mainly 4 biodiversity hotspots are there. That's the Himalayan region, Western Ghats Sri Lanka region, and Indo-Burma region, and Sundarlands. Why I am telling this point? Because the India is one of the very richest biodiversity areas. So they consist of these type of different types of tropical forests, that may be the tropical evergreen forest, semi-evergreen forest, mountain forest, grasslands, and other different kinds of the forest ecosystem that are occurring in our India. And mainly the India is one of the 12 megadiversity country in the world, and they contributed about around 45,000 species of the world, that covers an around approximate percentage of 8% of the global plant biodiversity. So especially in India, this global biodiversity contributed for 16% of the endemic species in the total whatever the well-known species of the plant groups. So among that, in the plant group, the phanerogames, broadly the plants are divided into phanerogames and the cryptogames. In the phanerogames, whatever the well-known documented species, the angiosperms are about represented by 19,390 species in India, and around gymnosperm is 81 taxa is reported in India. So when we take these phanerogames, they are well documented and well studied. But if you take cryptogames is generally referred as a lower plants. That's a non-flowering plant. Generally, we refer it as a cryptogames as a non-flowering plant that includes algae, fungi, lichens, mosses, bryophytes, pteridophytes. All these groups, these are known as a lesser known group of the organisms. They are very less or not get the attention of the scientists in a larger percentage. When we take what exactly these cryptogames, the first look at the classification of the plant. The Linnaeus has given the broad classification of the plant, that's the kingdom Plantae and Animaliae. So in his classification, he has mentioned around 24 classes. Among that, these cryptogamia is one of the very important class. So in that he mentioned algae, fungi, lichens, and bryophytes. When you take the Eclipse system, they separated it in the plant kingdom as a cryptogamic one of the group. Under that, he has also mentioned the thallophyte, bryophyte, pteridophytes. When we take all these group, we can look at what is exactly the biodiversity of each one group in Indian scenario. The first group, that is the algae. The algae are mainly characterized by the chlorophyll. They are the autotrophic organisms. They are largely distributed in a various habitat, that may be the aquatic or maybe the terrestrial algae. They are already reported. When you take the special characteristic, they are motile, unicellular in nature, and mainly these algae are nowadays they are mainly fuel resources and also the single cell protein resources. And this group of algae are in the world level, there are about 40,000 species of reported in all over the world. And especially when we take the India, there are about 7,400 species of algae are reported from 855 genera. And very interesting aspect is around 1924 is endemic to our country. And among these, whatever the algae reported, among that, if you take the terrestrial algae and freshwater algae, there are about 4,500 species of freshwater algae that belong to the 390 genera are reported. These fresh algae are the very good sources of single proteins also, and they are mainly the rich sources for the biofertilizers. And among these freshwater algae, the majority of them are belong to the chlorophyceae, basilarophyceae, and xerophyceae. When we take these basilarophyceae, they are generally represented as a diatomaceous earth. They are mainly nowadays they are used in the nanotechnology field for the production of different nanoparticles. So, and other group of the algae, that's terrestrial algae, around about 615 species are reported from India. And especially when you take the soil algae, there are about 1,600 species are reported from India, and around 880 species of marine algae. The very interesting group of marine algae, because whatever the marine algae, they consist of different group of the chemicals, that may be the agar-agar and gel, what we are using in ice creams. That's a very one interesting compound will be isolated from these marine algae. And nowadays, these marine algae are mainly used in the biodiesel production. So that's why it's one of the very interesting aspects of the study of bioprospecting in the algal field. So these are some common group of the chlorophyceae members and different chlorella and other volvax, Idogonium, different diatomaceous, that's the basilarophyceae members, and these are some chlorophyceae members such as chlorophyta and Pheophysa, laminaria, and glycosmosis. So all these different, and these some of the marine algae, that may be the sargassum, ulva, and this volvax, that's called as a colonial algae, and it's a single cell, that's a cladomonas. All these are the very interesting group of the algae, and these are some of the marine algae. They are commonly extracted for the different chemical compound. When we take the biodiversity of these marine algae is very interesting in India, especially the west coast and east coast consist of various different types of these marine algae is reported. The majority of them are the sargassum, amphilaria, and also the Leucaria. So these are the very interesting other group of the marine algae are reported from India. So this is about the first, that's the autotrophic group. The another group of that cryptogam, that's they are heterotrophs, because they lack the chlorophyll, and they especially they depend on the other plant, the saprophytic group of the plant. That's even they are saprophytic in nature and symbiotic because they lack the chlorophylls. That's why they depend on the other plant. That's another group that's called as the fungi. Generally we represent it as a group of mycota. Because these first group, that's they are autotrophs, that's they are synthesize their food, and this is second group, they are largely that's usually the symbiotic partner will be there, and also the saprophytic and parasitic type of the fungi we can be reported. Some of the fungi they cause the diseases to the plant, but side by side we have the larger economical values. When we take these fungal group, the majority of them are used in the production of some of the antibiotics, just like, and even they consist of chitin and other materials. They are largely used extraction for the different group of the manufacturing of different compound, especially the first that's antibiotic, that's a penicillin, extracted from Penicillium notatum. That's one of the wonder drug that's extracted from these mycota group. So when we take the diversity of these fungi, still now they are exploring more number of fungi from different part of the country, especially in the world. There are about up to now there are 97,000 species of fungi are reported from the world. They are belong to 8,000 genera, and India is represented by around 14,500 species. So belongs to 2,300 genera. When we take the total plant, this cryptogamic group, the majority of them are reported from the fungal diversity, because the various fungi causes even diseases, and some of the fungi are used for the extraction of metabolites, that's especially pigments, antibiotics, and also the other materials. And when we take the total biodiversity of these fungi, so in India there are about 3,000 species are endemic. Among that, the main group, that's broadly these fungi are categorized as ascomycetes, basidiomycetes, and deuteromycetes. Okay, so like that there are different group up there. Among that, the majority of them are belong to the in India, that is 1,600 species are belong to ascomycetes. They are represented by around 460 genera, and that is followed by that's the deuteromycetes and basidiomycetes. The basidiomycetes are very interesting group, because the many of the basidiomycetes used for the food, especially these macrofungi, that's the mushrooms, they are used for the edible purposes, and also they use it for the extraction of different metabolites, and they use it as anti-cancerous drug, and also used in the intestinal disorders. So like that, the various chemicals used in the different purposes. When you take the biodiversity of these macrofungal group, the mainly that's represented different group of, that's a jelly fungi, stomach fungi, wood fungi, fleshy fungi. These are the common name group name for the fungi, because some of them are, if they are grow commonly on the wood, they represented a wood fungi, or there are about 960 species are in India. That's mainly these, all these whatever the macrofungi, they belong to the group called basidiomycetes, because they produce a basidia as a fruiting body, so that's why they call it as a basidiomycetes. And even the jelly fungi are represented about 60 species. They consist of slime gel, and these slime gel used as a nanoparticles and used for the coating purposes, and puff balls, and also the sac fungi, that's a xylaria, that's generally represents deadman, that's a deadman fingers, and they are also used in the extraction of various metabolites. And these are the polyporous, geostram, and dictyophora, agaricus. All these are the larger group of macrofungi, and these are majority of them are used for the edible purpose and also the non-edible purpose. So this is one of the Amanita mascaris. This is one of the very interesting macrofungi. They use it for the extraction of that secondary metabolites and use it for the production of alcohols, because usually the Amanita consists of toxic substances also, but still they use it for the extraction of fermented product. That's a very interesting aspects. Okay, so the first two groups, that's autotrophs and the heterotrophs, and they have a special characteristic. And this is another group of the cryptogam, that's when symbiotic association between the first two groups, that's fungi and the algae, if they symbiotically association with each other, they form another one new group of the cryptogam or the plant group, or that is called as a lichens. So generally these lichens consist of two partner. One is a photobiont that consists of the algal group, and another one group that's a mycobiont. That mycobiont consists of the fungal partner. What you are gone through the first two groups, that's algae and the fungi, when they both the group are come in contact and symbiotic association with each other to form a new group, that's called as a lichens. These lichens are also one of the very interesting group of the organisms. That's a cryptogamic group. They are largely distributed. They are almost from the tropical to the tundra region, and they are of different growth forms. That's the crustose, foliose, fruticose, squamulose, and pendulous. These are the different group of the lichens. So if the crustose means they are tightly adhered to the substrate, and folios, they are loosely just like leafy like lichens are there, and fruticose, they are hanging in nature, or they are just like a pendulous structure will be there. And even the intermediate form, that's a squamulose and dimorphic form of the lichens are occurs on the nature. When we take the diversity of the lichens, around 20 to 8,000 species of lichens are reported from the world, and especially in India, around 2,956 genera of the lichens are species of lichens are reported from India. They are belong to the 79 families and 407 genera. So when we look at the lichen diversity and distribution patterns, when we take the each discovery pattern of this lichen diversity, in the earlier days very slowly they are discovered it, but nowadays from 2010 to 2020 large number of lichens are reported from India. This is one of the very interesting and attractive group of the researcher for the nowadays, because they consist of the secondary metabolites. So various secondary metabolites are occurs in these lichens. They use it for the antibiotics and pigments, and also they use it as a anti-cancerous and other antiviral properties. So that's why. And these are the some dominated families of the lichens families occurs in that's the Graphidaceae, Parmeliaceae, Physaceae, and Orthoniaceae. They are largely distributed. They are about Graphidaceae is represented about 431 species in India, and Parmeliaceae around 347 species, and like that Ramalinaceae and also the Telostaceae, Columetaceae. These Columetaceae, they mainly consist of cyanolichens. They are very interesting group of, they consist of the symbiotic partner as a cyanobacteria as a symbiotic partner. When we take the lichen diversity at the different states, the large number of lichens are reported from Uttarakhand, that is followed by the Tamil Nadu. That's the second one of the largest lichen reported state in India, that followed West Bengal and Kerala, Karnataka, and also the Uttar Pradesh. And even some of the states, there is no lichen reported from the Chandigarh and Diu Daman regions, and even Delhi. There is no lichens is reported from this India. When we take the different lichen geographical areas and the diversity of the lichens, that may be varies. Especially the Himalayan region, they are largely distributed, and this climate will more supported for the lichen diversity, especially the Western India Himalayas represented about 1,193 species, and secondly large distribution of the lichens occurs in northeast regions, and that is followed by the Western Ghats and also other central India and Eastern Ghats part. Recently the various lichens are reported from the Eastern Ghats and Deccan Plateau regions, that may lead to the future bioprospecting scope of these lichens in the Eastern Ghats also. And these are the different types of the lichens. That's the Pseudocephalaria, Parmotrema, Usnea, and Ramalina. And this is a pictorial representation. These are largely used in the different industry, especially the isolation of pigments and also the preparation of litmus paper. And also these are some interesting lichens. They use it in garam masala preparation. This Parmotrema and Parmeliana and these heterodermia, they are mainly used in garam masala, especially manufacturing these Hyderabad biryani or pani puri manufacture garam masala by these different group of the lichens. Their aromatic flavor will be there. So this is another group. So whatever the earlier group, that's generally we refer it as a talophytic organisms. And this is another interesting group of the cryptogan, a little bit advanced or evolved from the earlier groups, that's algae or maybe from the fungi. These bryophytes are very interesting group. Generally we refer it as amphibians of plant kingdom, because they require to water for the completion of their life cycle. When we look at the plant body, sometimes it looks like a green thalloid structure, but when we take the true bryophyte, they will have a sporophyte bearing structures, and roots and leafy-like structure will be there. And these bryophytes are mainly a plant body consists of rhizoids, and main thalloid structure will be there, and they are broadly categorized into three classes. That is a hepatic opsida, that's generally we refer it as a liverwort, and second one group, that's a anthoceropsida, that we refer it as a hornwort, and the third one, that's a bryopsida, that's a muci, that's a true masses. The first group, two groups are just like a thalloid structure will be there, and especially this hepatic opsida, name itself, that's a hepatic structure will be there. And they are even nowadays, these when we compare the doctrine of signature, these hepatic opsida, they are used mainly in the liver problem diseases. So like that, the medicinal value will be there for these bryophyte also. And when we take the different group of this bryophyte diversity, when we look at the diversity of the bryophytes in India, and it's around consists of 25,000 species in the world, and India is represented by around 2,562 species. Among that, if you take the each classes, liverwort is represented by 887 species, hornwort is represented by 39 species, and mosses, that is a true bryophyte, around 1,636 species are there. And the very interesting thing, that whatever the species he reported, the among that around 50 species are threatened category. So just we have to feel that before discovery of these species, so many species are moving to the threatened category. That's our bias of the studies. So still we need to be explored properly, and we have to be exploit their bioprospecting values. This will be lead to the future discovery of the various medicines and other things in the biodiversity field. And these are some interesting mosses are the true bryophyte, that's a Meteoropsis and Phalaenopteris and other bryome and Regalia. All these are the true bryophytes. And another last group of the cryptogamic that's represented by pteridophytes. Even the pteridophytes are smaller aquatic pteridophytes are there in the nature, and larger giant pteridophytes are, because these pteridophytes are a very interesting group of the cryptogam, because they occur from the Carboniferous period, and even so many fossil pteridophytes are reported in the world. And these are the generally we categorized in the plant. These are the vasculary cryptogams, because the pteridophytes consist of the vasculature, and they are generally occurs in terrestrial form, and some of them are epiphytic form, and some of them are xerophytic, aquatic, and even halophytic pteridophytes are reported. And the smallest, that's the azolla will be there, that's the smallest pteridophyte, and the larger trees for like cyathea and asophila. These pteridophytes are generally we categorize and into a fern and fern allele groups. The majority of these pteridophytes are, we refer it as a fern, and they consist of mainly sori will be there, and these sori produces the sorus. These are characteristic features of these ferns, and very interesting general characteristic, that's one is a frond. This is a young fern, young shoot of the pteridophytes. They are coiling in nature, and these fronds they used as a edible purposes, and when they mature they produce the sori on the beneath surfaces, adaxial surfaces, and they consist of the spore, and based on these spores these pteridophytes will be identified. In the world, there are about 12,000 species of pteridophytes are reported, and from India, around 1,293 species of pteridophytes are reported. They belong to the 206 genera, and among that, the 66 species are endemic to India, and already almost whatever the discovered species reported. So among that, around 211 species are threatened category, that is published by IUCN list in 2009. So when we take this diversity of these pteridophytes, they are distributed in various geographical regions of the India, that may be from the Western Ghats, Eastern Ghats, and Himalayan regions. The majority of the whatever the pteridophytes are reported, they are from the Himalayan region. Around 67% of the pteridophytes whatever the reported in Indian species, they are from the Himalayan region. Around they belong to 8045 taxas, and especially in the Western Ghats and Eastern Ghats is represented by around 345 taxas, and in Northeast region they represented around 340 genera. Among the whatever the reported species, the important families, that's a Polypoidae and Deuteroptidae and Atriniaceae. These are some interesting families of the pteridophyte. They are Polypoidae, that's represented by 135 species, and among the genera, that's the Selaginella and Diapteris. These are largely distributed. Selaginella is represented around 62 species. The Selaginella is one of the very interesting pteridophytes, because these are some group of the pteridophytes. It's a Xylotum, that's called as a rootless leaflet pteridophytes, and Equisetum and other group. Just coming to the point, this is a Xylotum, and this is a Lycopodium. And this is how the what I told, this is Selaginella. Selaginella is one of very interesting group of the pteridophytes, because if the water is not available, they completely dry it up, and when get the water again, they will be rejuvenate. We can call it as a Sanjeevani of this plant kingdom. So this nature is used for curing the various diseases. And equisetum, the equisetum is very interesting group because the stem region, that's the nodal region, they consist of accumulation of the silica crystals, and they use it as a extraction of the silica. And this is another one pteridophyte, that's the marsilia, that's a quadrifoliate leaf will be there, and they are characterized and used as the edible one. And this is osmonda, this is another one group of the pteridophytes, and it's a Dryopteris. And important aspect of these pteridophytes, majority of these pteridophytes are used as a ornamental purposes, and some of them are used for the edible and medicinal purposes. When we just summarize this total cryptogamic diversity, the largely group of these cryptogam is represented by that fungi. Around about 15 to more than 15,000 species of fungi are reported. Lichens are around 2,000, and around algae is represented by 8,000 species. And when we take the total number percentage of these whatever the reported cryptogams, when we compare their status, that's a very interesting aspects around whatever the reported cryptogams, that's around 30 to 40% are endemic to India. This is a great credited to the Indian biodiversity, because when you take the total biodiversity, how much of endemicity will be there on the biota or the flora, that is indicate the richness of the diversity of our continent. And the bad thing about this diversity of these organisms, just before the discovery of these cryptogams properly, they are moving to the endangered or threatened category because of the various human activities, that may be the increasing the population, habitat fragmentation, agriculture activities, or fire, that's a forest fire or human made fire, and also the pollution. They will impact on the diversity of these all group, and they may lead to the destruction. Just when you look at these pictures, generally the various threats will be there due to the diversity of all these, that may be the destruction activities, and also the cutting of the timber, agriculture activity. All these will lead to the effect of the diversity of all these groups, and the construction and over exploitation. This is a picture that's representing the how that much of the lichens are over exploiting in a particular regions, because the lichens for the small thallus lichens grow, it will take the several years. But when we are harvesting in this quantity, that may lead to the destruction of the particular group of the organisms. And this is how the over exploitation of these other groups, that lichen, pteridophytes and bryophytes, and they use it extensively in the market without the legal permissions. And these are the nowadays are taking some of the effort to conserve these cryptogamic biodiversity in India. They have taken the qualitative studies by the various scientists in the country, and creating awareness among the people about these economic value of these species, and what is the role of these species in the nature. Because when we take the lichens or the bryophytes, they are mainly indicators, forest health indicators. So that's why these groups are indicated as the status or the healthiness of the forest and the nature. So if they are luxuriantly grow, that indicate that the forest or the nature is in a good environmental conditions. So when we study these particular lichens or the bryophytes in a forest areas, if it is luxuriantly available, that indicates the particular forest region is a health condition, or the less pollution impact will be there on the particular nature or the environment. So, and taking the conservation effect or effort of Indian this cryptogamic biodiversity, the various wildlife sanctuaries and national parks are established to conserve this group of the organisms. And especially in NBRI, they taken the conservation, that's ex situ conservation. Generally, broadly biodiversity conservation in ex situ and in situ conservation. The in situ conservation are mainly by the wildlife sanctuaries and national park. In the ex situ conservation by the arboretum and botanical gardens. In NBRI, so various group of these cryptogams has established, and they started to conserve the arboretum in their botanical gardens. And even in the Uttarakhand, just recently reported shows that they started established a mass garden, and that's very interesting aspects. And these are the photographs of the first mass garden in the Uttarakhand region, that's in Nainital district. And this is some effort by the Malabar Botanical Garden to establish the lichen conservatory mechanisms, and they in the Malabar Botanical Garden they given a special area to conserve these Western Ghats lichens. And some of the lichen areas are represented as the lichen sanctuaries and even the bryophyte and cryptogamic lichen diversity areas. Just when we look at this overview of this presentation, the India is one of the richest mega diversity country and representing the various group of these cryptogams. Even the angiosperms are largely discovered from India, but when you take the cryptogams are very less known, but still they are exploited for more number of species and still need to be studied on this group, because when we take the bioprospecting values of the this group, that's algae or maybe the bryophyte or even the lichens and fungi, they are largely contributed for the economical benefit of the country. And even nowadays the various biodiesel and also the agar-agar and gel has extracted from these algae and other groups, they may contributed for the economic resource for the country. So when we do the biodiversity in proper manner and prospecting in a specific manner, we can get the more valuable bioactive compounds that may be useful for curing various upcoming diseases. And it side by side we have to think for the conservation of the all these groups, because when we exploiting in a more economic values, definitely there will be a threat on these groups. But when we look at the proper taking measures to conserve these group of the organisms, that will lead to the future proper conservation of this biota in our country. So once again, I'm very thankful for the organizers of these programs, and also I'm thankful to ENVS and NBRI for supporting data for our studies. Thank you, sir, for giving me an opportunity to interact with all these groups. So welcome any questions from the participants.
Participants, if you have any doubts, you can ask, sir. I think no doubts from participants, sir.
Yeah, good morning, Dr. Vinay.
Yeah, good morning, sir.
Good morning. This is Dr. Jagannathan. I am the next speaker to you.
Yeah.
But your presentation very nice and it's good. This is my different topic apart from myself. Okay, I have some doubt.
Yeah, sure.
In our Passion Institute, we have a lot of lichens.
Yes, sir.
Okay, here these lichens are placed for that to check the purity of the air. Am I correct?
What's that?
The indication of the air quality.
Yes.
Yeah, indication of air quality.
The indication of air pollution. Yes, sir.
Yes, our institute, it is started in 1904 for
Yes,
the
sir.
Passage of India vaccine protection. So it is started to the English people as per the French chemist Dr. Louis Pasteur. So there, in that area, at the time there is no pollution. Pollution-free area.
Yes.
So to identify the pollution system on those days itself, so more than 115 years itself, they are making small plantation. Each tree is having small lichens. Still, we have the lichens. Okay. Okay, here. But my question is, but nobody's from that, like cryptogamium, like other people from lichens working. They identified about what type of chemical indicator, what is the pollution indicator, and this kind of things. If it is possible, near future also you may come and see some of that specimens available in our institute for that other things. Okay, you can see from high mean sea level and also high altitude and this kind of things, but same kind of lichens we could not able to see in some of the terrestrial like Wanimadi or like that only. There are also some forests there. That is my small request to you, Dr. Nayak.
Yeah, thank you for your comments, sir. I hope this Pasture Institute occurs in a high altitude region. There may be possibilities of the various group of lichens, because especially these lichens are sensitive to the microclimate. So when they are having the good microclimatic condition, they are luxuriantly growing. And even these lichens are indicated by two types of pollution. One, some of the group of lichens are sensitive to the pollution. If the pollution percentage is increase the level, they will disappear. And some of them are accumulated the chemicals in the thallus, and we can estimate it to the UV spectrometer analysis, and we can estimate the how much of pollution percentage in the particular areas. That's a very interesting aspect, sir. You have observed very good things, and in the future also we can plan to work it out, or the group of the lichens and their bioprospecting values. Thank you, sir.
Thank you, man. Thanks to Dr. Nias for this kind of opportunity. What I asked.
Any questions from other participants?
Miss Meera, we have some short of time as other speaker is joined with us. You can proceed, Meera.
Sir, once again, I am very
Yes.
thankful to Nia sir and Agar Institute for giving me an opportunity to interact with you. And also, I am once again thankful all the participants to patient listening of my thank you. Sir, thank you very much, sir.
Vinayaga, sir, you have another responsibility. You kindly declare the best presenter.
Yes. After the sir presentation, I can tell the result, or now only I can announce the result.
As your wish, sir, you are going to retain. You can declare. If you have any commitments, you can.
Okay, just now I will announce the result.
As it is your wish, sir. You can declare at the end of Jagannath's presentation also.
Okay, yes, I will be there in the presentation.
Okay,
After
okay,
that, I can last the end of the sir presentation, I announce the results.
So, fortunately.
Now I would like to call our association president, Dr. I. Nias Ahmed sir, to give vote of thanks of this session and introduction to the resource person, Dr. Selvaraj Jagannathan.
Okay, thank you, Dr. Rasul Meera. It gives me immense pleasure, as one of the presenter is rightly pointed out, that a resource person with this high profile and with various portfolio with the expertise in knowledge, one of presenter is pointed out. Similarly, I go with his statement. We are highly fortunate to have Dr. Vinayaga with us. Even today is Sunday. After this program also, sir is wish to join with us. I really thank wholeheartedly for his availability. Getting high profile speaker for these kind of small NGOs is not an easy task, but I am highly fortunate. I am getting these kind of speakers with us. Even I am openly telling you that they are not claiming any kind of honorarium. Whenever we are doing some collaboration, we are mixed. In the other hand, I thank before introducing our another chief guest, Dr. Jagannath sir. Sir is joined very early and interact with Dr. Vinayaga sir and spoken about some collaboration. It is the need of the hour. Sir started the initiative. I really thank him. I thank other Agar members. When I got some feedback, there are more than 100 numbers of members in our association. There are a little bit participants are available. Then we discussed we have to go for some wide topic of webinar. Next Sunday, we have a speaker of Dr. S. Sri Ranjani Mokshagundam, Professor of Management, Sri Jagadguru Bala Gangadhara College of Management Studies, Bangalore. She is going to join with us. I will explain you the further details little later through the social media. Ma'am is going to enlighten us with crucial employability skills for educators. Tentatively, we are going to conduct this webinar next Sunday, similar manner. With this point, it is my pleasure to give the feedback of the presentation. Dr. Vinayaga give a detailed input about the characteristics of algae in aquatic as well as terrestrial. The fuel resources, SAR has given a output on SCP, single cell protein. He gave a statistics of the algae and the endemic nature, the biofertilizer application. In the other hand, SAR has enlightened us nanoparticle production. As I go with Dr. Jagannath's view, we belong to some other domain. As a researcher, we get interested about the nanoparticle production by using this algae and fungal material as a reducing agent. A graphical presentation of various slides. It gives a easy way to take some screenshots by various participants as a reference. The chitin production and penicillin production, a value-added productions are your detailed input, including the fungal diversity in India, the ascomycetina and other preval 25%, 40% ratio. He gives the images about the macrofungi in India. It is not an easy thing to see. The photos of the images are giving a representation of the various images, and the conservation measurement taken in the India, in the other hands, are giving a paper cutting of a live explanation. Instead of we are giving the article reference, if you give the paper cutting, it gives the various input about the information. I feel happy in one papers are depicted as Dr. M.S. Swaminathan, a very renowned agricultural scientist from Chennai. Sir is accompanied with, I think he might be Dr. Harini Kumar. Sir is one of my mentor from University of Agricultural Science. I feel happy to see the photos in his presentation. With this feedback, it gives me immense pleasure to welcome the gathering again, and it is my responsibility to introduce Dr. Selvaraj Jagannathan with us. Sir is joined with us little earlier as by our request. Sir is currently working as an assistant research officer in the Pasteur Institute of India, a historical institution located in Kunnur, Tamil Nadu, under Ministry of Health and Family Welfare. He completed his undergraduate and postgraduate degree from University of Madras in the subject of zoology and molecular biology, respectively. He aspired the following fellowship include our association, the fellow of Association of Global Academician and Researcher. He was a prominent fellow of Association of Indian Biology. He also a member in fellow in APSR, a NAPL certified assessor with ISO number in the biological pharmaceutical. He completed regulatory compliance in ISO 9001, 2000, etc. Sir is he started his career as a field assistant at National Institute of Nutrition, ICMR Hyderabad. Then he joined as a junior as well as senior research fellow in Department of Animal Biotechnology, Tamil Nadu Veterinary Animal Science University. Subsequently, Sir joined as a research assistant at Pasteur Institute of India during 2001. Now he promoted as a assistant research officer. He is having 4 patents and he published 33 research articles in various national and international journals. Sir has edited 8 books and authored 11 books and 8 conferences proceedings he participated. He aspired 16 awards for his credit from his UG level from his UG level itself. He was awarded with silver medal. He received the following awards: prestigious author in the year of 2010. Dr. APJ Abdul Kalam Young Scientist Award, Excellent Research Fellow Award, Best Researcher Award, Best Contribution in the Field, Dr. APJ Abdul Kalam Award for Scientific Excellence in 2020, Innovation and Discoveries Award and Distinguished Professor Award, Young Scientist Award in the year of 2022. Most Inspiring Award in the same year, 2022. Dr. Selvaraj Jagannathan, as a subject matter expert in the field of vaccine production, he is having 2.5 years of experience in R&D lab in life science, that is virology, bacteriology, molecular biology, along with more than 21 years of experience in vaccine production and testing bacteria and bacterial and viral. He was served as an editorial member in various journals. He served as chief guest, invited speaker, resource person, keynote speakers, chairing the session, etc. We are fortunate, as I affiliated with Sacred Heart College, or he's given physical expertise in physical presence in the webinar mode also. He has participated 131 webinars as resource person. While in the COVID season, I can able to remember wherever I go the social media, one, I get the invitation of Dr. Jagannath sir is presenting as a speaker. In another hand, some others contact me because I'm a good friend of Dr. Jagannath sir. Sir kindly recommend us to get his appointment. In the other hand, sir has gained 32 numbers of master degree. 6 number of M.Pharm graded, 21 numbers of BTech student, and currently 4 students are pursuing doctoral degree in microbiology under his guidance. He was a member and various committee as recognized in microbiology for MPhil and PhD, board of study member, industrial expert, doctoral committee member, technical expert in academic advisory committee member, central government member, institutional expert, external member in SME, examiner, and a estate committee member, library committee member, board of director, technical committee member, exhibition committee member, industrial training committee, coordinator WRD 2021, and he was also a member in the industrial safety committee. With this short bio, I feel happy and fortunate to invite Dr. Jagannath sir. I recused him. Kindly hold for a time. We have 3 presentation related to vaccine domain. Let them to present their views, followed by the resource person expert presentation. Thank you, sir.
Thank you, Dr. Niaz. And also once again, thanks to the all organizer and other things. I'll give a congratulations to the presenter. Shall I present my screen then?
1 second, sir. 1 second. Before we have 3 student presentation.
Okay, student presentation. Okay.
Ms. Kira?
Continue, sir. Sir, shall I call upon the participants?
Yes, proceed, ma.
Yeah. Mr. Shubham from Guru Nanak Dev University.
Yes, ma'am.
Yeah, you can proceed now, sir.
Okay, ma'am. Is it visible, ma'am?
Yeah, visible.
Okay. May I start, ma'am?
Yeah, you can start, ma'am. Proceed, ma'am.
Okay. Good morning, one and all. Myself, Shubham, from Guru Nanak Dev University, Amritsar. Today, I am going to deliver my presentation on vaccination and our body. First of all, we will discuss introduction. Vaccination, the act of introducing a vaccine into the body to produce protection from specific disease. And then, what is immunization? It is a process by which a person becomes protected against a disease through the vaccination. So, these terms are interchangeable with vaccination, or we can say that inoculation. Vaccination involves receiving a vaccine that is either injected through a needle or from drops into the mouth. Dr. Edward Jenner created the world's first successful vaccine. Now we move over to our next slide. Now, what is principle of vaccination? Vaccine contain antigen that stimulate the immune system to produce an immune response that is often similar to that produced by natural infection. This black figure indicates before vaccination, white figure indicates during vaccination, and colorful figure indicates after vaccination. Next one is types of vaccination. There are several types of vaccines like inactivated vaccines, mRNA vaccine, toxoid vaccine, viral vector vaccines, live attenuated vaccines, subunit recombinant polysaccharide conjugate vaccine. One by one, we will discuss briefly. First one is inactivated vaccine. This is a vaccine consists of virus particle, bacteria, or other pathogens that have been grown in culture and then killed to destroy disease producing capacity. Second one is mRNA vaccine. It works by introducing a piece of mRNA that corresponds to a viral protein. Third one is toxoid vaccine. In this, we use a toxin, or we can say that harmful product made by the germ that cause disease. They create immunity to the parts of the germ that cause a disease instead of germ itself. It means the immune response is targeted to the toxin instead of whole germ. What one is viral vector vaccine? A viral vector vaccine is a vaccine that uses a viral vector to deliver genetic material that can be transcribed by a recipient's cell and to elicit an immune system. Last one is live attenuated vaccine. In this, the pathogen is killed and remain active in live vaccines. However, it is attenuated, or we can say that modified in a way that the pathogen is not able to cause disease itself, but can produce robust immune system. Now we will discuss how vaccination works. Different type of vaccines work in a different ways to offer protection. But with all types of vaccine, the body is left with a supply of memory T lymphocytes as well as B lymphocyte that will remember and protect the pathogen against disease.
Proceed, man.
Shubham sir, have you completed? Sir?
Now we move over to importance of vaccination.
Sir, go to the zone.
Ma'am, what ma'am?
Yeah, go to the conclusion soon, sir. Only 5 minutes
Oh,
allowed.
okay, ma'am. There are 2 to 3 slides more.
Yeah.
Vaccination protects you by creating an antibody response without you having to experience potentially severe illness. Is there any side effects from vaccination? Studies reveals that there are minor side effects from vaccination. It includes pain at injection site, fever, fatigue, headache, muscle pain, chills, and so on. At last but not least, we will discuss protection against vaccine. Vaccine protect against many different diseases, including cholera, diphtheria, hepatitis B, measles, mumps, rubella virus, Zika virus, influenza, tetanus, smallpox, and so on. At last, I will conclude my presentation. Our immune system or sounds the alarm so our body knows there is an infection. Then our body releasing antibodies to fight the germ. Antibodies as soldiers designed to fight off the specific germ you have. The antibodies work to attack, weaken, and destroy the germ. These are the references from where I am taking my content. Thanks for your kind attention. And also I would like to thanks to the organizer for giving me this wonderful opportunity. Thank you one and all.
Yeah, Shubham.
Yes, sir.
Yeah, it's a good topic as your master degree level.
Thank
Okay,
you, sir.
I have 2 basic questions.
Okay, sir.
What kind of antibody generated once pregnancy mothers are vaccinated?
What's your what?
What kind of antibodies
Yes,
during
sir.
the birth of the baby while that baby's mother vaccinated during pregnancy?
Sir, it's a passive.
It's a passive.
Yes, sir.
Are you sure it's a passive or acute?
Sir, passive.
Passive immunization means what?
Sir, passive vaccination.
Are you know or not know?
Not, sir.
Okay. Okay, passive immunization. Okay, I will clear the question's answer now. So,
Okay.
baby, the mothers during pregnancy is vaccinated. So, the
Okay.
developed antibody transferred to the baby via placenta. At the
Yes,
time
sir.
of born, the baby got acute immune response. Okay, that's answer. And another one, passive immune response means already secreted antibodies to the person. That's a passive immune response against active. Okay.
Okay, sir.
Okay, good things. You may call next person.
Thank you, sir.
Thank you, sir. Now I call Inbavalli ma'am from Acharya University, Institute of Allied Health Science. Inbavalli ma'am,
Yes, ma'am. Yes.
you can start now, ma'am.
Ma'am, not able to share the screens. Can you help me out?
Nia sir,
Miss Inbavalli, what is your name shown in the webinar?
Inbavalli, sir. Recent advances in
No, that's not my question.
okay.
We can't able to track you.
Surveys it is there.
Okay, surveys. That's I'm asking you.
Yes, sir. Yes, sir.
Now you can present.
But it's showing host disabled participants.
No. Now we try. Now I make enable.
Yes. A very good morning, one and all present here. First of all, I would like to thank you to giving me the opportunity, wonderful opportunity to present my thoughts of this about vaccination. So I'm going to take my topic, recent advances in vaccine technologies. In previous person, Mr. Subham already presented about vaccines. So we know about that vaccine. Vaccine is a biological preparation and that improves immunity to a particular disease. Okay, so I'm not going to detail about things, these slides and at all. We know about the history of vaccines from 80th century to 21st century. So many vaccines are found out and cure the so many diseases also. So in 21st century, currently, 2019 and 2020 to 23, we were affected COVID-19 vaccine. That is one of the main important vaccine.
Excuse me, I'm sorry for the interruption.
Yes, ma'am.
Make the screen zoom. Full screen, you can show it.
Okay.
May move to slideshow. Just click the slideshow. Move to the slideshow. Then it will take slide from current slide.
Slideshow.
From current slide.
Is it okay, sir?
Yeah, it's good.
Okay. So that is what COVID-19. So that is one of the important vaccines we were found out from in our India. That is very proud moment. Then coming to this under development, so many vaccines is going to be implemented. So some kind of vaccines, that is cytomegalovirus vaccine and Epstein-Barr virus vaccine, hepatitis C vaccine, herpes simplex vaccine, HIV vaccine, and respiratory syncytial virus vaccine, Zika virus vaccine, Lyme disease vaccines. Then the latest technologies in vaccines, that is one of the newest latest technology, that is mRNA vaccines. This mRNA vaccine is going to deliver detailed instructions to the body cell to make key protein antigens. Then recent advances in vaccinology are the disease like Alzheimer's, stroke, and therapeutic cancer vaccines. And also in UK, the children are affected in meningococcal conjugated vaccines. These are all the vaccines in recent vaccinology is going on under the development. And several types of vaccines, as Mr. Subham presented over that. So I'm going to skip this types of vaccine. Now I'm going to move on, need to implement. See, nowadays, so many peoples all over the world, so many peoples are affected, diabetes, tuberculosis, renal disease, arthritis, then bone related disorders, back pain, women health, thyroid, PCOD, UTI, breast cancer. So I'm going to discuss about these things in my mind recently. From birth to 18 years, we are having the immunization schedule. But after 40 years, we didn't have any immunization schedule. In the middle, after 18 to 40 years, we were be healthy, as all the people were be healthy. But after 40 years, we are affecting these kind of diabetes, renal disease. That is the main important thing. And coming to this women health, so many women are affecting thyroid, PCOD, UTI, and breast cancer. We don't have any vaccine about these kind of things. Even coming to this diabetes first, I'm going to tell about the diabetes. We are having n number of research paper available in diabetes. We know the root cause of diabetes. We know everything about diabetes. Why don't we go for that vaccination about this kind of diabetes? That I don't know. So many people are affecting. This is a slow diseases to kill the persons. So many n number of persons all over the world affecting the diabetes. They are struggling to face the life to living the, that is what they are difficulty to feel. So many number of, there are n number of surveys are also available in diabetes. Nowadays, even kids also having diabetes. That I don't know why we are having these kind of things. Why don't we go for the vaccine to this kind of diabetes, these diseases? Then coming to this tuberculosis, that also that a number of persons affected tuberculosis, there is no vaccines. Vaccines is available, but will it healthy? Will it helpful or not? I don't know that one. In my surveys, actually, in present of us, I saw many people affected the kind of tuberculosis and get so many related diseases also they are affected. Then coming to this renal dialysis, after nowadays renal dialysis is going on a very improvement way, I think so. So many patients are going for renal dialysis, even small aged people also going for renal dialysis. They are struggling to treat this renal dialysis. That also, that is one of the important thing we want to, in my mind, it is showing, asking me, why don't we go for this renal kind of diseases, vaccines also. Then coming to this arthritis, the n number of people above 40 years of age and 70 to 80 years of age, they are affecting arthritis and bone related disorders, back pain. In survey says that in 10:8%, they are having the back pain. Because of that back pain, they are troubling their work, whatever it is, the family, take care of family, everything. So these also one of the important, we can go for the vaccine to find out these kind of things. Now coming to this women's health, in women's, there are many n number of women's affecting thyroid. So why they are getting thyroid? Hormone imbalances. So why don't we go for this to cure the hormone imbalances to our body? So that also my thing is going for that to find out the vaccine of this thyroid problem, PCOD problem. Then PCOD problem, the n number of girls also affected. Then UTI, the n number of surveys is going on that about this thyroid, PCOD, UTI problems is going on that. In our research paper, it said the number of papers available in Google Scholar.
Ma'am,
So then,
excuse me. Can you go to conclusion, ma'am?
Yes, ma'am. 2 minutes.
Okay, ma'am.
Then even breast cancer also, the number of women are going to die because of this breast cancer. So why don't we hope to implement to introduce above the 40 years of immunization schedule? We can go for this one. My wish is to all the people should live without any complicated disease up to at least 80 to 90 years of age. So as I have to be concluded this point, active people is a healthy nation. I quote this one, healthy citizens are the greatest asset any country can have. Health of the people is the health of the nation. Health for all must be the fundamental goal of all nations. Thank you once again to giving me the opportunity to present my thoughts over here. Thank you, sir.
Yeah, welcome, ma'am. It is an informative one.
Thank you.
Now I call upon Aisha Sabine, ma'am, from
Yes,
Madurai.
ma'am. I'll be sharing my screen.
Miss Meera, sir wants to interact.
Yeah. Yeah. Yes. What's the name? Your presentation is very well, but you ask something about.
Yes,
So first, I clarify about your questions.
yes, sir.
Okay. First question you asked about meningococcal vaccine. You mentioned
Yes,
the meningococcal vaccine only at UK. It's a
Not
conjugate.
only UK, sir, but it is present in.
Yeah, in India also we have that vaccine that is from
Yes,
that conjugate. And another things you asked about vaccination about. So we are not having any vaccine after the 18 years. So we
No,
have.
sir.
Yeah, you
After
mentioned up to 18. You
Okay,
mentioned that only up to 18 you only few vaccine. There is no vaccination against other age people. And another thing, we have the vaccination that is called as adult vaccination. So adult vaccination, it's around TT is there, and also influenza is there. And what are the vaccine preventable diseases that vaccine we may going to have being as an adult. And another one is I will clarify that for BCG. So for
Yes, sir.
tuberculosis, we have the vaccine. That's a childbirth vaccine. At the time of birth, we are vaccinated. But here, that vaccine is from Mycobacterium bovis. It's from cattle-based strain only used for the BCG vaccine that was developed by Galmet and Kurin. But what diseases currently we have in Mycobacterium tuberculosis infection to the all, that the entire diseases are caused by the other strain. There's a various strain. There's a strain divergent only. The TB, we are not, could not be vaccine preventable. So that's why other peoples are making the vaccine using it for disease conjugate and also Angara-based vaccines. So a lot of us going on for that to prevent the TB, ma'am. And another thing you mentioned about diabetics. Okay, for the diabetics, you are the biochemist girl, my lady. That diabetes is a metabolic error. Okay, here that is not caused by the any microbial, not from a bacteria, not from the fungal, not from the viruses. That is a microbial, that is a metabolic error on which technology we can use to the development of vaccine. If you know means, let me know that we may go further at that one. Okay, and the other thing, yeah, these are the things only, my things, probably. Okay.
Yes, sir, about tuberculosis, there are no awareness, I think so. No awareness about this vaccine and at all. In even village people are affected, these kind of things, they don't know.
Yeah,
so what is the vaccine?
we have. If you're moving to any government hospital, we have the RNTCP program, revised National Tuberculosis Eradication Program, and also every hospitals, every PHC, every small village hospital, they have the separate thing about TB. Lot of
one of my village person, he is affected tuberculosis for long years.
Okay.
He went for treatment in government hospital also, I don't know. After long years, now he has a problem to not able to walk, and something happened in his leg and hand and at all.
Yes, ma'am. Actually, we have the no. So every treatment and diagnosis, everything free of cost in all GH and everything in the district headquarters and other things. Once a person having the continuous sputum and also the chest infection, other thing, that person he has to easily diagnosed by the GH people. So GH also they have the under the treatment for the quarantine. Lot of drugs available. So, as per your report, there is no much more awareness. Awareness will be taken by the government policies only, the state government, other things. Okay, that is my view, ma'am. But anyway, it's a good initiative.
Thank you, sir.
Once again, congratulations.
Thank you, sir. Thank you.
Okay, next, Aisha Sabin.
Yes, sir. Good afternoon, sir.
Good afternoon, please, ma'am.
Sir, is my screen visible?
Yeah, it's there. Go with full presentation.
Yes, sir. I'm sharing.
Ma'am, share with slideshow, ma'am.
Yes, ma'am. Yes, ma'am. Searching also.
Okay.
Yes, ma'am. Is it clear?
Visible, ma'am.
Yes, thank you. Good afternoon, all the dignitaries, participants who have joined virtually. I am here to share my presentation on COVID-19 vaccines. From the past 2 to 3 years, we have been attending enormous webinars, seminars, enormous sessions, focusing only on this pandemic COVID-19. So I'll brush it up quickly. This COVID-19 is a disease caused by a virus called SARS-CoV-2. It spreads through contact with any other person who has already been infected with this virus. And it is predominantly a respiratory illness. It can affect other organs as well. And the symptoms, the incubation period range from 2 to 14 days. And the major symptoms include fever, chills, cough, shortness of breath, fatigue, muscle aches, headache, loss of taste or smell, sore throat, congestion, nausea, vomiting, and diarrhea. Coming to COVID-19 vaccines, the two major vaccines which ruled the pandemic were Covaxin and Covishield. Covaxin was India's first indigenous COVID-19 vaccine developed by Bharat Biotech in collaboration with Indian Council of Medical Research, ICMR, National Institute of Virology. This vaccine is actually an inactivated vaccine, and its efficacy was nearly 79 to 80%, and the storage is 2 to 8 degrees, and doses were 2 doses. Whereas coming to Covishield, this was developed by Serum Institute of India, Pune, with contributions from AstraZeneca and Oxford University. And this is a non-replicating type viral vector. Efficacy was overall 70.42, and the storage temperature was 2 to 8 degrees Celsius, and it was a regimen of 2 doses with a gap of 2.5 to 3 months. And there was a large debate we could see with our people, with our family members, with our colleagues, with the society, that which vaccine is better, whether Covishield was better or Covaxin was better. There is nothing like any one of these is better. There is no direct scientific evidence that, and we cannot compare. Each one has its own efficacy, and as per vaccines, there were some side effects and which were highlighted to a very above level. And there is no proper comparison between these two, and we cannot like say this one is better or that one is better. No, both these vaccines helped in preventing the infection as well as preventing any severe illness. We can choose any of these vaccines subjected to the availability, and we have already chosen. Many of us have done with all the both doses, and we have even gone to the further doses. Right, next. Coronavirus vaccines in India. Apart from Covishield and Covaxin, we have some other vaccines developed in India, and India is considered as a vaccine hub. I'll tell you why. And we have some, only this Covaxin and Covishield, these are ruling the market, and we have some other vaccines like Corvabex, which was cheapest and economical. It is a recombinant protein subunit, and PTX COVID-19B, it was India's first mRNA-based vaccine, and it is a 1-dose COVID vaccine. And we had Zycov-D. It was developed with support from Center for National Biopharma Mission. And we had Sputnik V in an activated SARS-CoV-2 virus. It is also 1-dose variant. And vaccines by India Immunological. This is an innovative codon deoptimization technology which directly targeted the cells infected by SARS-CoV-2 and was Bangalore-based Mindvax. And for kids, we had Covaxin and Bharat Biotech's 1-shot nasal vaccine, Zycov-D, and Serum Institute Novavax, which is termed as Covavax, for kids. Right. Before having vaccine, there were a lot of prejudices, lot of issues before having, like who should have a vaccine. Only if a person who is healthy, he should have a vaccine. Any person affected with disease or any other health issues, they shouldn't have vaccine. And before we have vaccine, to our healthcare provider, and we should know some our health issues. We should know if we have a severe allergic conditions or anaphylaxis to any drug or any food or any vaccine, it should be informed. If we have a fever, we should inform them. And if there are any bleeding disorders, or we are taking a blood thinner, or we have received any other COVID vaccine, it is not mandatory to receive another vaccine. And if we are immunocompromised, or if a person is pregnant or planning to become pregnant, all these should be informed to the healthcare provider before taking the vaccine. And these COVID vaccines, no doubt, they were successful in protecting humans from this deadly pandemic, and it brought back the country back to its original position like before. And these vaccines help to reduce the risk of the pandemic, again, coronavirus. And it is important to make sure that people have access to the vaccination without any discrimination and aware of the benefits of the vaccine. And there were a lot of myths about this, about taking this COVID vaccine in our own community itself. Like COVID vaccine is not a safe vaccine. It makes people infertile, and this may result in severe side effects or even death. And I do not need to wear a mask if I get vaccinated. These vaccines can weaken the immune system. This will alter my DNA. It is unsafe for pregnant women, and it may cause long-term negative effects. And this virus has muted, and no longer this vaccine is going to work. Keeping aside all these myths, I would like to conclude that with the vaccine, when we are taking a shot of vaccine or we are taking a dose of vaccine, we will be definitely getting rewarded with necessary protective antibodies that will be fighting against this deadly coronavirus in particular, or any other virus in general. Thank you for the opportunity given. Thank you.
Wonderful presentation, ma'am. Sir,
Yeah. Yes. Aisha, ma'am.
Yes, sir.
Okay, it's a nice presentation, a very good topic.
Yes, sir.
Okay, and also let them clear some, I have some doubts.
Okay, sir.
Okay, you mentioned about, just go to a slide about COVID vaccine in third slide.
Wait a minute, sir. This
Yeah,
one,
that's slide. Yes, sir, that's slide. Go back, another slide. Go back.
this one.
Yeah, this one.
Yes,
So in this slide, you mentioned about developed by Bharat Covaxin and Covishield.
yes, sir.
And here for Covaxin, you mentioned you written as a N bar A.
So no, that's what I mentioned there. 79 to 80%. It was a previously taken pick, whereby efficacy was not mentioned. I tried to change, but sorry, I prepared at the last minute. I mentioned, sir, while I was talking.
Yeah,
I
you
mentioned, but I couldn't change that.
Yeah, as I know. But here, where you got this information?
Sir, actually, one of yes, sir, I'll be clear. One of my friend is a doctor. She works at Haryana, Rohtak Hospital, Haryana. She was my close friend, one of my classmates.
Okay.
And we were discussing, like, even I, as personally, as a citizen of India or as a human being, I had lot of doubts regarding the vaccines, Covaxin, Covishield, and we used to discuss, like, which one is better. I used to consult her before getting vaccinated. Which one should I get with? Whether I should go for Covishield or Covaxin. I had a lot of discussions with her. And she assured me that she'll be sending me a gist of what's going on in the field of this vaccination. Like currently, it was like before 1.5 years maybe. And we were discussing, and she provided me with the data, sir. Her name is Dr. Jessim.
Okay.
She works at Haryana.
Anything, you were from the faculty of a particular college, MTKC. But here, any presentation we are going to give will reference something. Even our final slide, but I expected, but it's not, you mentioned that your final slide.
Sir, I didn't get you, sir. What?
Yeah, you are not mentioned the references.
References, okay, sir. Right.
Yeah, that things. And also this kind of information is clearly present in our Ministry of Health and Family Welfare website. There is a dashboard of MOHFW.
Okay, sir.
Other thing, this kind of details also mentioned that World Health Organization COVID-19 dashboard.
Okay, sir.
So that also we can mention that.
Right, sir. I'll follow
okay.
it.
Go to that and next slide. And not next slide. Another slide. Yeah, here. Yes, this one. Oh, you mentioned about Corbevax vaccine.
Yes,
that is not a Corbevax vaccine, it's a Corbevax.
Sir, Corbevax.
But yes, it's a Corbevax.
Corbevax.
Yes, Corbevax is from biological events vaccine.
Yes.
Okay, again, go to the final slide.
Right, sir.
Okay, final slide. Okay.
Yes, I'm coming.
Yeah, it's here. Okay. Here, okay, COVID-19 vaccine successful protecting, no? You mentioned COVID vaccines are successfully protected human from coronavirus to make the country back to the original position like before. This is not mentioned in our Indian government organization. It is not from the ministry. declaration.
Okay, sir. Okay.
Okay, still we have the corona.
Yes, we have, sir. But
Corona.
we are quite back to normal and we are still fighting with the
Yeah,
pandemic.
we are
Yes,
normal.
yes.
Our body is also normal,
Yes, sir.
but
Yes,
corona it is an abnormal. Okay,
yes, sir, definitely, sir. Yes,
that's
sir.
based on a lot of strain variants there. These are the things there. So these are the things near future also. You can mention and detail. So I present based on the report was published. I present based on the report was mentioned in the dashboard of. So kind of like that.
I'll definitely include, sir. Yes, I
That
missed
is
it.
good
I'll
for
be
you.
doing it.
You know, it's not a mistake. It's a
No, no, sir. Like I prepared it in a very short time, sir. Like I wanted to present, but I couldn't. I'll rectify
Yes.
it, sir, in my
Good,
further
good, good.
presentations.
But
Right,
anyway,
sir.
it's a good. So
Thank you,
once
sir.
again, congratulations and all
Thank
the
you,
best
sir.
for your
Thank you, sir.
future presentation.
Yes, sir. Thank you, sir.
Okay, thank you.
Yeah, wonderful presentation.
Thank you, ma'am.
Sir, I request you to take over the session, sir.
Yeah, it's good. Shall I present my screen? KS?
Yeah, you can share it, sir.
Once again, good afternoon to all. Yeah, my slide is visible.
Not yet, sir. It is starting.
This is starting now.
Yeah. Yeah, it's visible, sir.
Yeah, okay, good. Yeah, it's okay. Once again, good afternoon to all the presenter, all the participant, organizer. Thanks to Dr. Nias, the great fellow. So we are that every initiator of that entire biological sciences, AGAR, and what kind of person Dr. Nias is an highly motivatable to the student. It's a good motivator. I inspired him from that various aspects. Okay, like vaccine. So vaccine also highly inspired to us. So in the first slide, you can see that various type of images. So one is representing about medical field. Another is the great man Louis Pasteur, and the next is the AGAR. Because of AGAR only we are associated. And another thing is here the technology. First, you can see this technology. So here this technology is various kind of technology, vector-based, adjuvant-based, synthetic vaccine, under the production of vaccine production process unit, and next to the vaccine. Now I'm moving to the my first next slide. Here is a vaccine. What is meant by vaccine? It's a biological preparation that improves immunity to a particular diseases. Typically contains an agent that resembles a disease-causing microorganism. Often it's made from weakened or killed forms of the microbes, like toxin or any surface proteins. That agent stimulates the body's immune system towards a recognized, like a destroy it or keep as a memory cells. Here, the small mice, like us people, the vaccine introduces safe amounts of antigen. So here, safe means that antigen should not cause any pathogenicity. That antigen should not cause any advergency. So that's why I mentioned a safe amounts of antigen to the body. So we have the immune system. That immune system is going to recognize the immune system. Further recognition that moving to the development of antibodies, either T cells or B cell based antibodies. T cell and B cell based antibodies, some of the primary immune response, that antibody is going to neutralize the antigen. Some of other antibodies is going to keep in as a memory cells. Sometimes it is in a plasma cells or memory B cells. That differentiation, the resisting memory cells and the antibody secreting plasma cells, it keep in various aspects. This is how vaccine and vaccine immune response. Right. So here, there is a antigen is not only a vaccine component. So under the other important component, it's here. One is active ingredients, that is antigen, and adjuvant like ALUM, aluminum phosphate gel, or lipid nanoparticles, and also we have the M59 and oil in water emulsion. That adjuvant help in boosting the immune response to the vaccine. That is the enhancement of antibody level. And other thing is the antibiotics. It prevent contamination by a bacteria during the vaccine processing, like neomycin sulfate. That neomycin sulfate it using for the especially on the viral vaccine production to keep the material in uncontamination based. So moving to the stabilizer. For example, sucrose and human albumin, maltose. So these are the stabilizer to keep the vaccine in stable during manufacturing and maintain its effectiveness during storage. Because vaccine production process, it is not from single plus 22-degree temperature. It's ambient. It is started with minus 197-degree temperature as a seed. Then moving to the plus 4-degree temperature, it's a medium. And moving to the 37-degree temperature, cell culture. Then again moving to the plus 22-degree temperature, it's ambient. Again moving to plus 4-degree temperature for harvest. Then moving to the minus 20-degree temperature for the material. So various kind of temperature, that antigen, that active ingredient, it will be in stable. It should be stable. So that's why we have added some stabilizer like sugar or gelatin base. Then moving to the preservative. Once a vaccine was prepared and formulated, once it's open to the vaccine, for example, some multi-dose like flu vaccines or DPT vaccine, it should not be able to under contaminated. So to avoid the contamination, the thiomazole is the agent. So we are keep the vaccine in a preserved condition. Next, the other trace components like formaldehyde, ultraldehyde, and beta-propylactons. So these are the chemicals used to convert the toxin to toxide, convert the inactivation. So these are the these are the trace elements. Everything is going for removed. Everything going for clearly vanished out by the purification aspects. Before the vaccine formulation, all the ingredients are tested and the safety, and only is going for the vaccine supplier. Here, you can see that one. There's a history timeline of the most important vaccine. Here, the history timeline important of the vaccine. Before 18th century, there's a smallpox by the great man Edward Jenner. In 18th century also, the 19th century, typhoid, plague, rabies. In 20th century, starting with diphtheria, pertussis, tetanus, tuberculosis, influenza, yellow fever, and rubella, mumps, measles, polio, tick-borne encephalitis, hemoplex influenza, hepatitis A and B. Now, in the 21st century, meningococcal D, pneumococcal vaccine, and the rotavirus, zoster virus, herpes, human papilloma viral vaccine, and the COVID-19 vaccines. So these are some timeline is the most important the vaccine industry. Here, moving to the type of the vaccine. Live attenuated vaccine is a primary vaccine. So altered or weakened form of the microorganism which lacks virulence but retains the ability towards create immunity against the diseases. For example, oral polio vaccine, chickenpox vaccine, measles, mumps, rubella vaccine. Then inactivated vaccines, a live organism are killed and then further inactivated and used as vaccine. They usually eat stable and do not need exact temperature storage. For example, rabies vaccine, pertussis vaccine, and the co-vaccine. Then moving to the toxoid-based vaccine. Some of the bacteria, it can able to release a toxin, either exotoxin or endotoxin. That toxin we are collected or harvested. That toxin further converted to a oxide by the detoxifying chemical like formaldehyde or glutaldehyde. So that is a toxide-based vaccine. One is a tetanus toxide vaccine, another is a diphtheria toxide vaccine. Then moving to the another category is subunit, recombinant, polysaccharide, and the conjugate-based vaccine. And here, and mRNA also is there. So these are produced by employing more modern technologies by purifying and isolating the active components of the microorganism which produce immunity but not the diseases like hepatitis B, HPV, and also in COVID-19. Here, I'll give some status about available vaccine. That is a 26 vaccine preventable diseases as per World Health Organization. So some of the pipeline vaccine is under development stage, not in the up to the level of releasing. One is a cholera, COVID-19, dengue, hepatitis, influenza, hemoplex influenza type B, and human papilloma viruses, influenza, then Japanese encephalitis, malaria, measles, meningococcal, mumps, pertussis, pneumococcal, poliomyelitis, rabies, rotavirus, rubella, tetanus, tick-borne encephalitis, tuberculosis, typhoid, and varicella, yellow fever. These are the available vaccine as per the World Health Organization. So another category is under pipeline vaccine like enterotoxigenic E. coli, Group B Streptococcus, HSV, herpes simplex virus, HIV-1, and malaria, so Neisseria gonorrhea and non-typhoidal salmonella type disease, norovirus, paratyphoid, and respiratory syncytial viruses, schizometasis viruses, and Shigella, Group A Streptococcus, tuberculosis, and improved influenza vaccines. Yeah, so once we find out the vaccine, we're moving to the vaccination. So the major contribution that vaccines have made, saving countless lives around the world. There are 27 causative agents against the vaccines are available, and many more against targeted for the development of vaccine. The vaccine not only for preventing infectious diseases, some help the body fight range to illness by the activating the immune system towards the recognize and attack the diseases. Vaccination is one of the most beneficial and cost-effective measures we can adopt to protect ourselves from the potentially fatal diseases. Every year, up to 4.5 million children's lives are saved by the immunization, as per the World Health Organization. Here, vaccination is proven and one of the most cost-effective child survival interventions. All countries in the world have an immunization program to deliver selected vaccine to the targeted beneficiaries. And the next is a universal immunization program. Minister of Health and Family Welfare, they have another that is called as in our India, we are following that, especially on that. Pregnant women, infants, and children who are at high risk disease of preventable diseases, that is a UIP. So all countries are having their own immunization program. So to target on newborn baby as for the pregnancy mother. So that is free of cost. So this kind of vaccine like DPT, BCG, hepatitis B, TB, rotovaccine, JE, Hib, measles. So these are the vaccines under UIP, and JE is commonly known as brain paralysis and meningococcus, and pneumonia is particular on the particular state only. So these are free of cost. Okay, here why the pregnant mother advised to take a tetanus toxoid to overcome the neonatal death. 30 years back, a lot of pregnancy mother they faced the problem in the means of problem in that a lot of the babies, newborn babies, going to die. So finally, traceable that the Clostridium tetani spores forming bacteria, it is present in that scissor and pulses during the labor surgery of delivering the baby. So that kind of spores, it directly affects the newborn baby through the respiratory infection and other things, so leads to the death. To avoid that, so while the month of 5 and 7 in the pregnancy of the mother, the vaccinated, the developed antibody, it's IgG, it's a transfer to the placenta to the baby. At the time of birth, the baby got acute immune response. That response is called a tetrastructured antibody. That antibody is going to neutralize the exposing that antigen. Yeah, here is how the vaccines are development. One is a prototype pathogen approach. What is my prototype? It's a working model. Pathogen means it's a disease-causing organism. How it is going for identification. For example, I mentioned only in the viral groups. So we can divide the virus in based on the structure, based on the function, based on the molecular level. So structure means icosahedral, it's an hemisphere or it's a bullet shape or it's a spherical. And also another pathogen selection is based on the function like Bunniaviridae, Hepavidae, Enpaviridae, and the Rhabdoviridae. So a lot of virus family. And another thing is a DNA and RNA based. So there is a pathogen. So here during the pathogen selection, whether it is caused by the outer membrane, either caused by the inner protein, or it's caused by the other nucleotide base. So this is going for, we underwent for the basic preclinical and translational research. So this is a combined research, interdisciplinary research of the pathogenesis and the immunobiological and the structure function. Then moving to the, based on that, we are going to the assay development. So based on the assay development, that reagents development and the pathogen biology, so we are using with the animals model, that is in a translation research. So when the translation research moving to the clinical trial to the phase 2, either we are moving the monoclonal antibodies for the immediate treatment or moving the vaccines for. This is a prototype pathogen approach method for the vaccine development and other. Here you can see that how the vaccines are developed, it will take minimum 12 to 15 years. 12 to 15 years processes. And here, the 12 to 15 years processes, first the pre-PCT and the ACT, academic clinical studies, phase 1, 2, 3, and 4. During the PCT, the establishment of the vaccine candidate has the desired effects in vitro. So here, in vitro means as in the laboratory condition. That effects produced consistently. Here, consistently means it should not be deviated from the one value to another value. Then, once the very good results we obtained from the PCT, then moving to the animal studies. In the animal studies, we are selected the animal model, like there's a mice, or Swiss albino, or guinea pig, or is going for rat, or is going for rabbit. So here it's a protector to ensure about the immunogenicity. Then moving to the human being is a healthy volunteer under the phase 1 studies. So here only we are identify about the dosage calculation, then safety assessment using with blood samples. Then moving to the phase 2 is 100% volunteers, around it is in 1,000 numbers. So this 1,000 numbers volunteers are based on the age and the intended, as well as this phase level, we are checking about that additional safety and efficacy tests. So here, during the stage, those who are affected with the particular diseases, that affected person only going for vaccination in a phase 2A and a 2B. And moving to the phase 3, after the successful result from the phase 2A and 2B, there's a 1 lakh, around 10% to 1 lakh volunteers, and establish a protective immune response and a clinical consistency, or obtain the very good results in the safety aspect, safety, potential immunogenicity aspect, that vaccine going for marketplace, and moving to the phase 4. Here is a phase 4 is a pharmacovigilance. In the pharmacovigilance is post-marketing surveillance and detect the adverse effect, assess the long-term efficacy. So these are the very broad roadmap towards the development of new vaccines. It will take minimum 5 to 12 years. And moving to the next slide is vaccine development. So these are what I mentioned, what are the common gaps, why it will take a lot of time towards the vaccine identification development. One is a lack of foundation knowledge in the basic research. Here, surface structure and viral replication, entry mechanism. Here, if you identify some pathogens, then how the surface structure, whether it's made up of carbohydrate, or made up of the protein, or made up of the lipid, or made up of any other common molecule, and viral replication, whether it's a viral replication what's happened inside too, or it needed any other gene response, then I'm going for receptor mechanism. So that entry and receptor mechanism is to find out, so where is going to entry through the alveolar cells, or it is a cell membrane, or outer cell membrane, or any trafficking mechanism was there. So due to the lack of foundation knowledge, and also another aspect and lack of small and large animal model and the resources, based on the pathogenesis and reverse genetics and reagents based that, so we may choose the animal model. Sometimes that animal model, mice, it will be give a much more immune response, but it's not exported from the primates. But sometimes primates give a very good response, not in the mice. But some of the vaccine, it may give in the mice as well as the primate as well as a very good in potential level, but it not is in human being levels. So that's why it's a lack of very appropriate animal disease modeling, and also host response and mechanism of operation. And another gap is quick scalable platform technology. One is during manufacturing technology and medical countermeasures. That's it. A biochemical product technology, it's a cumbersome process. So while making of a fermentations and making of a media preparations and making of other things, so a lot of gaps is there. So once we coordinate, and once we come across that entire things, only the vaccine going for manufacture are going to produce that. Okay. So anyway, we have developed the vaccine. Then how it's going for scale up? In laboratory scale, in our R&D level, in college level, or university level. So it can it produces at research and early development stage. It's around a small size of manufacturing, like 100 to 1,000 times less than production scale. Used to support formulation and packaging development, early clinical and preclinical stages, and also we are analyzed to assess the evaluation of a CQA as a critical quality attributes. So same process we are going to scale up in the pilot scale level. So here is an 1,000 liter to around 500 ml to around 5,000 liter. So use the process development and optimization, the stage to support preclinicality and a mid to later stage and clinical evaluation, and also to support formal stability studies. Here, stability study means to check the vaccine capable and how long it's going to expire, that in which temperature that antigen is going to stable, and which temperature that antigen stability is going to get down. So these are things only we can update from the stability studies. And also scale should be relevant and to be discussed with a concerned regulatory authority. This balanced parameters such as anticipated product volumes, anticipated site of the production, equipment constraints at the site, and the regulatory expectation. Here is a regulatory expectation means, that is why GMP means CGMP, current good manufacturing practices. For example, if you have the small fermenter in your lab condition, but that fermenter having small indicator or sensor anything, but same type of sensor, it will not applicable for them tomorrow or day after tomorrow, because that regulatory has changed, keep on changing the entire regulatory aspects. That's why it's a regulatory expectation we should have to fulfill, and also for that other good laboratory practices. Then moving to the production scale. So these batches are of the size that will be produced during the routine manufacturing and marketing the product, and also it needed for phase 3 and PV stages. So PV stages is process validation stages. So process validation stages, once the instrument will be, the design is a design qualification. The same instrument we went to give you in that company that is in a factory acceptance test. So after receiving the instrument or equipment, we are fixing in our department. There is the installation qualification IQ. After the IQ completed, then going for performance qualification or a running method. So after the PQ, then going for operation qualification. Operation qualification leads to the process validation method. So various qualification of each and every instrument towards the certification. Okay, here there is a vaccine development, process scale up, moving to the vaccine production. So here is a vaccine production. In what facility we have? I have very few slides. One is HVAC, heat, ventilation, air condition, for that clean air environment and entire surfaces area. And water, we have that raw water, pure water, and also water for injection. Per day we need 150,000 liter water for the production aspects. And also electrical 24/7 compression plotters, and the CA. CA means compressed air and a chiller and ETP, STP system. Towards that, keep the entire environment in each and every condition, and also not having the viable and non-viable particle inside our facility area. Then this is our inside the facilities area. You can see that entire facilities area is a fully screened, and one is a control panel and seed fermenter. HF4 is there, 2 seed fermenters there, and 2 batch fermenters there as a control panel and laminar hood. So everything is interconnected. And again, moving to the formulation and filling. Here formulated with the entire group of vaccine under the clear condition, and moving to the filling and final processing. So this is a vaccine production facility what available in our institute. Each and every vaccine, it should be tested in the animal. How can we test it? We can choose with some animal. Here we have the Swiss albinomyces. That Swiss albinomyces are more compatible with our DPT group of vaccine. What we have produced here, like potency, immunogenicity, and also that antigenicity, and also identity. So various kind of studies we have carried out this animal model. So these animals is approved by the Institutional Animal Ethical Committee as per CPC's guidance. And along with, we are following that IP, Indian Pharmacopoeia regulatory norms. So how that regulatory norms are declared. So based on the regulatory norms only, we are fulfilling our this thing. And another images you can see this only intracerebral and also intraperitoneal, there is intramuscular typhoid injection. So these are the approved method what we are following in our institute as a vaccine testing. Okay, here is a bacterial vaccine production. You can the two small kid, there's having his jaw, their entire spinal cord look is a bow movement. This is caused by the Clostridium tetani. It's a spore-forming bacteria. It will anaerobic environment. It is present that all over the world and all the places. Once it will enter to our body, it needs anaerobic environment. It leads to that this kind of things. This is a neonatal day of the children. To avoid that, in our institute, we are the production of DPT group of vaccine from the 1962 onwards. So that Harvard strain, here our strain is a vaccine strain, is a non-pathogenic and also immunogenic, highly immunogenic as well as non-spore forming bacteria. That Harvard strain, we are reviewing in a ATG medium, alternate thioglycolate medium, and check the purity. Then intermediately, we are moving into HIGB or inclusion growth medium. Then moving to the production, we have the fermentation using Muller and Muller medium around 1,000 liter fermented. We are working in around the 700 liter working volume. The 700 liter working volume, we kept the culture in a 7 days process. 7 days process, we have supplemented the spore aeration as a surface with the carbon dioxide because it's anaerobic and also around 600 RPM and also it's maintaining 37 degree temperature. So entire processing on the day of 5, that Clostridium tetani bacteria, there is no non-availability of the media sources. That enter bacteria under for the suicidal activity. Through the suicidal activity, the bacteria are ruptured. That is ruptured and self-suiciding, release a toxin called as a Clostridium tetani toxin. So that toxin again moving for the upstream downstream process is moving with the clarification using 0.45 and 0.22 micrometer filtration to avoid the contamination. Further, the sterile toxin underwent for the IPQC testing. In process quality control testing like LF for ml antigen level and minimum lethal doses and multiple toxin value and also protein nitrogen content. These are the purity aspects. With the sterile toxin underwent for the detoxification process by added with formalin is 0.6%. So 4 days in room temperature and then further 4 weeks in a 35 degree temperature. Then we collect the sample after the detoxification tested in an animal to check the irreversibility of the toxin. If it is not a if it is a toxide, then moving for a crude toxin sublot. Then moving to the concentration, that higher volume around 700 liter material, finally with the concentration we reached only 7 liter by the 50x material using with the 30 kilo Dalton of the cassette system based on the nominal molecular weight limit of the membrane protein. So here after the concentration, it's moving to the salting and salting with a secondary purification aspect, ammonium sulfate fractionation. So that after the ammonium sulfate fractionation, it's moving for the final filtration using check with the bacterial and fungal sterility and antigen content, specific toxicity, antigenic purity, irreversibility in a guinea pig, and that is called as a BPDT. That means bulk purified tetanus toxide. That BPDT sample we sent to the QC quality control department. They also reanalyze the safety, efficacy, potential aspects. Once our entire vaccine fulfill the entire criteria as per the regulatory norms under the guidance of quality assurance, they are taking the sample through the QCD along with the results we obtained from the QC department. We sent to that National Regulatory Authority. It was located at Kasoli as per IP guidelines and WHO guidelines. They all also reanalyze our vaccine stability and efficacy and other things. So once our vaccine fulfill that entire criteria, immediately the same vaccine going for the release to the nation. So these are the bacterial vaccine production, especially on the tetanus toxide. Here, moving to the viral vaccine production. Here, the viral vaccine production is entirely different from the bacterial. So bacterial only we need a fermentation culture, a media, single seed. But here, the virus seed. So viral, we need a live cells. So live cells in percentage we are using on cell line that is called as a VRO, African green monkey kidney epithelial cell line. It's a propagated in a continuous manner. You can see that monolayer culture virus cell line. Here, there's a media. The cell culture media reagents, monolayer virus cell line, propagation cell substrate. So once formation of monolayer, then going for selection suitable viral seed. That's the fixed viral seed. Here we have the virus seed that was originally developed by Louis Pasteur. We will have a virus strain. So that strain only going for infector. So these are the rabies virus structure. You can see that. So here the rabies virus structure, then moving with the rollerboard culture, Then after the infection, every 72 hours, we are collecting the viral harvest. That viral harvest then pooled, it's called as a single viral harvest. That single viral harvest, so going for the virus concentration by the downstream processing. By downstream processing, using with inactivation, using with appropriate inactivating agent, you can see that what you call is an TFF system. That cross-flow filtration, that material 200 liter viral orals material by the fermentation process here, biorate of this thing. So it's moving to that 200 liter. Finally, we reached only in 5 liter of material. So based on 10x concentration, that 5 liter material again going for affinity chromatography purification. So after the purification, so here, by the purification, they are using with that suitable purification method by affinity chromatography. So collection of the elution fractions at the entire fraction to check their safety, efficacy, potency, antigen content, and also host cellular and non-host cellular DNA, non-host cellular protein. Here we are using with the viral cell line is a host of that propagation. So that final vaccine, that the DNA of the viral cell line and also that protein of the viral cell line should not be present in the vaccine material. So everything we are removed through the affinity chromatography method. So then moving to the formulation using with suitable stabilizer, additives, filling, and fridge drying aspects of filling. Then moving to the fridge drying. So the entire material under the freezing in a filling condition. After filling, it's moving to that vaccine sample by the QC people. That QC peoples are taken the sample and check the results. The same results we are sent to that serial Kasoli. The Kasoli reanalyzed our safety, efficacy, potency as per our results. Once everything's a correlator, the same vaccine going for the release to the nation. So these are the production process of virusul-based rabies vaccine. It will take minimum of 8 months. So in the future vaccine technology, using with artificial intelligence technique, you can see nowadays also we have the non-viral technologies there. Using without viral protein, is a pathogen viral protein. So in future it has to come. I thought this is my last slide. Yeah, this is the more I study nature, the more I stand amazed at the work of the creator. Science bring man nearer to the God. The Louis Pasteur, as a question hours. Okay, these are intro. So thank you. Any questions you may ask now.
Such a wonderful presentation, sir. Very informative one. Hello.
Yes, I'm hearing. Yes,
I would like to call sir now, our association president, Dr. Niaz Ahmed sir, to give vote of thanks. And declaration will be done after the vote of thanks given.
Okay, thank you, Dr. Rasul Mira, for chair the session very good manner. First of all, I would like to thank our resource person of this session, Dr. Jagannath sir. Jagannath sir make it more interesting by asking questions at the end of the session by each student. It makes more interactive. At the same time, I will call it as a brainstorming session because sir interacted with more questions with the faculty members. It makes them very wide discussion. It will go reach the students. It is a trick sir might happening. I understand from his various presentation as a listener. He interacted with Ingoli ma'am and Aisha ma'am. Was very nice interaction. It was a technical correction as per the Ministry of Health policy. Sir, you pointed out very informed policies. The faculty members are well taken the points. I really appreciate them. In the other hand, sir has presented here while you are talking about the highly committed resource person. They are designing the PowerPoint with include very recent research updates. You can see the presentation of Dr. Jagannath sir, including the slide number and including agar logo, then CFD, FDP, header and footer. Honestly speaking, his background also very nice to see. It makes us to more motivational for our faculty members. As Dr. Rasul Mira talked to me, our organizing secretary sir, he was very loud to the subject sir, as per the interaction he did with the various presenter. Vaccine and immune response are give a detailed note on the common vaccine components, active ingredients, and the vaccine presentation. preservation agents. It is somewhat new to us, preservation agents, history and timeline of vaccine production, what is the prototype and a pathogen approach, pregnancy-related vaccines, and SAR give the detailed note on clinical trials and translational research, because it is new to us. We can't able to see the human trials. 15 years of process, because we can't able to know as it is a human research. It's about the roadmap of new vaccines. It includes rat, rabbit, and the human healthy volunteer. The numbers of sample taken from 1,000 volunteer to etc. etc. Various phases are give a detailed note on phase 1, phase 2, phase 3, and phase 4, from lab scaling to pilot scaling and the production scale. You give a various subheading vaccine production facilities available in their laboratory. It is central government laboratory. My wholeheartedly tell you, while we are trying for the industrial visit to your institution, I talked to many times with Dr. Jagannath sir, but our head of the institution is not permitting because it is far away. But at the same time, I feel happy we can show this presentation. It's a virtual IV, virtual industrial visit. We can able to see various things happening in the PAST Institute, how the vaccine testing is going on, animal experimentation, institutional animal ethical committee, and Indian pharmaceutical regulation. So how the normal viral vaccine production takes place till the end of future vaccine technology. So he were detailed input. He were detailed a diploma course in vaccines. Really happy to be associated with Dr. Jagannath sir. His availability, I wholeheartedly thank him on behalf of the association for his availability during the Sunday. Thank you, sir. Thanks for your availability.
Thank you, sir. Once again, I thanks to the entire organizer and also that entire member. So keenly, they are step by step your coordination with all aspects. Okay.
Thank you, sir. Now I call Dr. Vinayaga to declare the presentation, who is did some good presentation. It is not race 1 and 2. Whatever the thing you are thinking that he is good presenter means, you can declare the result. We will dispatch the merit certificate. Vinayaga, sir.
Yes, sir. First of all, I congratulate all the presenters. They have presented very nicely. For the just declaring the result, just I will announce first and second presenters. The first good presentation is given by VR Pradeep from Savita Dental College, Chennai. So once again, I congratulate Pradeep for the nice presentation on the topic that's related to the role of antioxidant vitamins. Congratulations, Pradeep. And second prize is goes to that Miss Huraya Lakwi Ahmad Khan from the same that Savita Dental College, Chennai. She has also presented very nicely on the topic of evaluation of anti-inflammatory potentials. She has presented very nicely, and once again I congratulate both the presenters for the nice presentation, and others also. They have shared very good informations regarding their present research topic. So I thank organizers for giving me an opportunity to interact with you, and also I thank Jagannath sir for the nice presentation and good information talk on vaccination. So thank you, sir. Thank you very much.
Thank you, Dr. Vinayak.
Thank you, Dr. Vinayaga sir, for joining with us. Now the turn is Dr. Jagannath sir.
Yeah, I have three presentation. Actually, it's a good narration. One is how the vaccine and immunization. Next is what is the recent advancement in vaccinology, and so moving at that COVID-19 vaccine. So what I mean, my slides also presented like that only, but I like that guy is Subham. He's in master degree level only. So he gave various information, say mRNA-based vaccine also that things. So that kind of person will give some information. And also another thing, in Beverly, she also spoke well, and she is a recent advances in vaccinology. I think she upgraded entire knowledge about what are the vaccine kind of technologies available in these aspects. And moving to that, Aisha Sabina, and she was infected. And currently, the COVID-19 based vaccine details, no one take a risk about their vaccine details presentation. But first, my heartfelt congratulations to Aisha Sabina for getting the first one, and moving to the Inboli is in second. and as well as these things. And other things very nice presentation and a very good topic chosen by others. Once again, congrats to the entire AGAR team and other aspects. And thank you once again. Thank you. See you in other FDP in which will other thing. Bye, sir.
As again and again put a thank for your availability, and I would like to thank my friend Dr. Vinayaga also. Thanks for your availability and make the webinar a grand success. Thank you one and all. We will get the certificate as early as possible. Thank you.
Thank you, sir. Bye, sir. Take care.
Thanks, sir.
Okay,
Meera, we can wind up, ma.
Okay, sir. Other participants